I welcome all the dignitaries to come up on the stage. I request all of you to give a huge round of applause. Sir, please get seated. Thank you, sir. Good morning, esteemed dignitaries, distinguished speaker, pharmacy professional, researchers, and bright young minds. Welcome to Day 2 of National Conference on the theme Integration of Pharmacovigilance and Regulatory Affairs for Enhancing Drug Safety. I am Rushika Jaiswal, Associate Professor, your host for the day. I am absolutely delighted to see such vibrant crowd once again. Now, I would request all the dignitaries to come over for Saraswati Vandana. I request our student Meenal Devtade to sing Saraswati Vandana. Pause, उनको वहाँ जाने दें.
या कुंदे दुतुषार हार धवला या शुभ्र वस्त्रावृता या वीणावरदण्ड मंडित करा या श्वेत पद्मासना या ब्रह्माच्युत शंकर प्रभृतिभिः देवी सदा वंदिता सा मा पातु सरस्वती भगवती निशेष जाड्या पहा निशेष जाड्या पहा.
Thank you all the dignitaries. Thank you Meenal for singing the beautiful song. I request all the dignitaries to come over the stage and get your place. Before we jump into today's schedule, let's take a quick look back at the incredible insights shared yesterday. We had phenomenal session starting with inaugural ceremony. Our conference was enlightened with esteemed professional from the pharmacy background. Chief guest Dr. Abhay Itarwar sir, Principal Guru Nanak College of Pharmacy, and Dr. Ajay Pise sir and Dr. Shrikant Phule sir. Yesterday's esteemed speaker were Dr. Alpana Asnani ma'am, Professor Priyadarshini JLC College of Pharmacy, Nagpur. Her topic was life cycle and safety management of the drug. Next session was delivered by Dr. Ajay Pise sir, Associate Professor, Dadasaheb Balpande College of Pharmacy, Nagpur, and his topic was Startup in Pharmacovigilance. Third session was delivered by Dr. Shrikant Phule, former Drug Inspector, FDA India. His topic of discussion was Historical Background of Pharmacovigilance. Last session was delivered by Dr. Omji Porwal, Director of Academic Research, Quaivan International University, Suleimania, Iraq, and his topic of talk was Integration of Pharmacovigilance and Regulatory Affairs for Enhancing Drug Safety. The interactive question answer session showed just how passionately our community is engaged in solving the current challenges in pharma sector. Today promises to be even more exciting with the focusing on day 2. Today's esteemed speakers are Dr. Kashyap Nagaria sir, Assistant Divisional Head, F&D, Otsuka Pharmaceutical Private Limited, Ahmedabad. His topic of discussion will be regulatory implication and strategic roadmap for pharmaceutical formulation. Next session will be delivered by Mr. Chinchulkar Sir, Senior Regulatory Affair Associate, Paraxel International India Safety Services Private Limited, Bengaluru, Karnataka. His topic will be strengthening regulatory framework for integrated drug safety oversight. Third session will be delivered by Dr. Darshan Telange, Associate Professor, Dattamage College of Pharmacy, Wardha, Maharashtra, and his topic will be regulatory and pharmacovigilance challenges in nano-enabled drug delivery system. Last session will be delivered by Dr. Jignesh Doshi, Director, Center of Interdisciplinary Research and Development, GH Raisoni University, Sai Kheda, Madhya Pradesh, and his topic will be AI in pharmacovigilance from reactive safety to predictive intelligence. Now, moving towards the facilitation ceremony, I request Dr. Rupesh Duje sir to facilitate our today's guest, Mr. Shubham Chinchulkar sir. Now I request Shabnam Sahar ma'am to please felicitate Dr. Dr. Darshan Telange sir. Thank you, ma'am. Thank you, sir. This event is not just a meeting of mind, it is a collaborative platform to catalyze change in healthcare through innovative research and sustainable pharmaceutical practices. Moving on, our today's speaker needs no introduction. They have left an incredible mark on the field of pharmaceutical science through their pioneer research in the field of regulatory affairs. I request all the pharma buddies, please give a warm welcome to Mr. Shubham Chinchulkar, Senior Regulatory Affair Associate, Paraxel International India Safety Services Private Limited, Bengaluru. Sir will extend his view on the topic strengthening regulatory framework for integrated drug safety oversight. I request all the dignitaries to get off the dais. Sir. Mr. Shubham Chinchulkar, sir, is experienced regulatory affairs specialist with 8 years managing complex regulatory end-to-end submission and compliance for international authorities including EMA, MHRA, US FDA. Sir motivates to support business goal by ensuring the regulatory excellence and collaborating with high performing team in growth focused environment. Sir deals with work like CMC regulatory strategies, risk management related to regulatory activity and GMP inspection. Sir was a part of Intas Pharmaceutical Limited as assistant manager. Sir completed his MTech from Naipur, Mohali. To Sir's credit, there are many research and review papers. With this briefing, I request Sir to start with the session. Sir, please grace the stage. Thank you. Thank you.
Hello. Hello. Hello, am I audible to everyone? नाही चालेल चालेल हां चालेल थोडं खाली या कोणत्या काय करायचं चालेल आहे का देऊन दे ना हां तुम्ही चालेल So, good morning everyone. Good morning to Principal Sir and all the delegates present here. I am very privileged to be invited here as a keynote speaker for this national conference. And without wasting time of everyone, I'll move to the point directly and will brief about my industrial experience and as well as the bridging between the industry and the academics. Yeah, so today I will be talking about this strengthening regulatory framework for integrated drug safety oversight. Don't look at the headings directly. It looks little complicated, complex, but it's not as it. It reflects itself the meaning that I will be talking about the regulatory framework which help us to maintain the pharmacovigilance activity efficiently and which ensure the patient safety among all. That we know, we pharmacists, it's our first ethics to take care of the health of patients and medicines. So that's what I selected and choose this topic to talk over and provide some glimpse because it will not possible to talk over this topic in an hour, but I can give you some glimpse so that it can help you to bridge your gap between the academics and industry. So let's move forward. So the flow of my presentation will start first with the introduction, then the importance of drug safety, what would be the current challenges, the case for strongest framework, global harmonization, then leveraging technology, patient-centered regulations, future outlook, and conclusion and call to the action. So complete presentation of mine will be playing around these keywords like patient-centered regulation, framework, transparency, compliance, accountability, and AI. So we'll start with the introduction first. So regulatory affairs itself means it will control and it will regulate all the medicines flow into the market. So it will directly ensure the patient safety, medicines effectiveness, and high quality of the medicines as well. And it also, regulatory affairs will act as a bridge between the industry and regulatory authorities. So this is very critical and important for the drug approval and the life cycle management into the industry. So it will also play an important role to ensure the compliance and global regulation, because without regulatory affairs and rules regulation, the medicines would not be much efficient into the market for the patients. It also supports the pharmacovigilance activity. Because of that, the various submissions and various regulations will be managed. Then guidance for the preclinical studies, we know in academics also, when we're doing some experiments, so we do it on the rat, mice, guinea pig, rabbits and all. So this all comes under the preclinical activity. So regulatory will not only manage the clinical, but they also look into the preclinical activities. Then approval of all the clinical trial protocols, ethics and everything will be also taken care by the regulatory. Then final conclusion and the final point would be the IND and CT. IND is the investigational new drug applications, and the clinical trial application to all Europe and US will be submitted, and to ensure the smooth approval workflow. Then first and important point of presentation would be the importance of drug safety. So it should be generally comes into our mind that what should be the importance of drug safety, because we we know that recently also we saw the case in the market of the cup syrup users in the Africa that took the lots of cases and lots of life of a child. So that's what the drug safety is much important into the picture. So drug safety is the foundation of our public health, because without drug safety we cannot use any medicine and it will harm the patient's health and everything. So for that, the WHO and IMA emphasizes that safeguarding patient is the primary objective of the regulatory framework. So whenever the regulatory comes into the picture, it directly relates and relevant to the patient safety. Then the relevant guidance for this issue is ICH 210, and the related guidance highlight that the drug safety is not limited to the approval. It also needs to be required throughout the life cycle of the drug product. It should not be the case that once you approve your drug, your responsibility is over. No. Until and unless drug completely get accredited or terminated from the market, till that moment you have to work as a regulatory professional to maintain the life cycle of a drug product. Then internal regulatory framework for this would be the European and FDA, like GVP and the REMS, risk evaluation and mitigation strategies. So what are the current challenges and why we need to look it into very seriously at this moment? So current challenges would be fragmentation. If we see, there are lots of countries and lots of regions in the market. We know majorly the EMA and FDA because they are more stringent and they are very controlling. But other than them, regulated market, semi-regulated and rest of the world is also there, just like African countries, American countries, Israel, CIS region and all lots of things. So according to that, guidelines are very differing and they are having the different standards. So that's what this is an important constraint regulatory flow, that the guidance has been fragmented across the globe. Then the delayed detection. What is the another point? Then weak data integration slows the response of safety signals. So it means whenever if any adverse reaction has been noticed out, it not usually go to the health agency very rapidly because of several gaps. We'll discuss it in the upcoming slides. So because of that also the safety signals received very lately, delayed, and it impact the patient safety. And the last is the evidence gap. The real world patient data underutilized in the decision. So whenever if we consider the example where two drugs are marketed into the country like India. So if both are the same from the different innovator and different marketing company, if one drug shows some effect, then the second company should adapt that and they should overcome that effect and adverse effect. But it is the gap that they do not show or do not see the available evidences into the market that what drug should cause and what drug should have harm on the patient. So this is also another challenge. Because of that, the patient safety is getting compromised nowadays. Then the case for stronger framework. So what should be the possible way out to overcome such challenges and to cross these hurdles? So integration is the key. This is the biggest point we can see. Integration, it means that there are two meanings. It should be regulatory and pharmacovigilance should get integrated in a proper way so that we can achieve our objective and ambition. Another thing is integration of all the guidelines from all the regions so that we can get the single guideline to propose our medicine into the market. Then the strengthening of regulatory framework, like if we see the diagram, then first would be the pharmacovigilance system governance. So pharmacovigilance system should be very firm, well established and well versed so that we can avoid such hurdles. Then risk management planning. Whenever we submit or whenever regulatory authority or whenever some industry submits the molecule to agency, then there should be a proper risk management planning. Then adverse events reporting. Whenever any market recall happen, any adverse effect happen to the patient of certain population, then that should be reported to the authority within a time frame. Signal detection and management, safety communication, risk minimization measure. These are the case for stronger framework. So we'll not definitely go through this because it is high level information. So just to brief you, so these are the domain where we need to look into. If you see the left side column, which is in the bold, system governance, risk management planning, adverse event reporting, signal detection management, safety communication, and risk minimization. So according to the countries, there are classification, like first column is dedicated to EU, that is European Union, where 28 countries are there. Second column dedicated to the US FDA, which is the most stringent in the world. And third is the WHO, is the World Health Organization, and it runs for all African countries. And African countries are the biggest market for regulatory of any company. So these are some guidance, like system governance should be done according to the GVP Module 1, PSMF. For US FDA, it's a different 21 CFR Part 314, and for WHO, the guidance is a different. So these all are respective guidelines according to the domain. Then next would be the global harmonization. So this is the important part where we can achieve our objective. The global harmonization is nothing but the process where all the regulatory authorities come into one panel and they decide and they map and they outline the guideline considering all the variations of the population and the available and existing guidance. Then it also should narrate the guidelines for development, approval, and the monitoring. Development will consist of all the R&D activities. Approval will consisting the market, the product comes into the market, and monitoring will be all the pharmacovigilance activity, right from the risk management plan to reporting the adverse effect and everything. Then what would be the goal of this harmonization? Unity. So its goal should be strengthen oversight of medicine by creating unified framework that ensure consistent safety standard globally. Then what would be the scope? It covers the registration, which is the initial one, the clinical trials like phase 1, phase 2, phase 3, everything, phase 4 as well. Then pharmacovigilance activity, then manufacturing quality, and the post-market surveillance. Then for this, there are lots of union and unities are present, but we'll talk about only few which are very crucial and important. So among them are the first one is the ICH, is very well known to everyone who already fascinating towards the regulatory, which is ICH, International Council for Harmonization, then the WHO, FDA, EMA, and PICS country. So PICS I want to emphasize here, maybe you are aware about the ICH, WHO, and FDA, and EMA, but PICS may be very new to you. So PICS is a pharmaceutical inspection cooperation scheme. It is the organization, or it is, you can say, one firm which looks over the good manufacturing practices for all the nations. So they have their own specific person guideline, single guideline, which need to be followed when we need to go to some countries like African and American countries. So your manufacturing facility should be PICS approved. Then only you will be accepted in the African countries. Yeah. Then what are the benefit and impact? So for the United States, reduces the duplication of clinical trial and animal testing. This is the currently very demanding field. Then faster access, patient will get the faster access because of this harmonization. Cost efficiency, improved pharmacovigilance, and global trade. Yeah. So then what are the challenges for the harmonization? The first would be the partial adaptation across the countries, because we need everyone have their own standards and they do not want to show flexibility because of their population situation and population nature. But still there is a scope, we need to little show the flexibility to adapt the practices and provide the patient safety. Then regulatory sovereignty concerns, capacity gaps in developing nations, because developing nations are not much developed, so they always face some technical problem, they don't have the technical staff, so all this administrative work also impact these things. Then difference in the implementation scope, transition cost for industry, it is a well-known factor which impact the harmonization scheme, because to make a new guidance, to make it harmonized across the globe, it's also very costly because it involved the review and approval process from the lots of SMEs which situated or seated in the EMA and FDA. So they are very costly. So this is also another impacting factor. Then emerging therapies oversight like gene therapy and everything. Data sharing and confidential issue, because agency usually not get ready to share their data because of piracy, because of their marketing competitiveness. So this is also the impactful factor. Then legal and administrative barrier. Then There will be the case example, will not explain much details as we already see. So case example is AMRHI is Applicant Medicine Regulatory Harmonization Initiative. So in harmonization, majorly impacted market is African countries because they are very backward now also. So that's what majority of organizations are looking towards the African countries as their potential market. So if we see the harmonization parameter, then African market will definitely come into the picture. So this AMRHI is established in 2009 and supported by the WHO, and their work is that to harmonize the guidance according to the people population and according to the guidance. Then objective is same, to improve the safety efficacy of medicine. Focus is harmonization. Mechanism is collaborative network between the medicine registration and priority given to the essential medicines. And then impact will be the definitely faster approval because less gaps, less differentiation, less distinction, so faster approval will be in our hand. So there are a few organizations and the initiative, AMRHI, just we seen, ICH, PN, DRH, SN, and APAC. So next point will be the leveraging technology. We all know we are living now in the era of AI, where everything is done and happen with the help of AI. But I just want to emphasize here, AI is important, but along with that, humans are too important. Because recently I can tell you one case, I think it's 2 days back, one of the major organization in the globe where they have done the manufacturing process records, which is BPCR, based on which the medicine has been prepared into the company. They have written this document with the help of AI, but they did not check thoroughly. So FDA caught them and FDA provided them, shared them the 483. Because of that, their manufacturing has stopped. Since that, FDA mentioned AI is fine, but you have to have your control as a human being. Till that, you should not use the AI because AI also quote last statement with asterisk mark, should not use this information directly without verification. So AI is important, but the person who drives the AI, it is the most important. So if you want, do not to get replaced from the industry, then adapt the AI and learn how to act smart with the help of AI. So we'll talk here about the leveraging technology. Here we can see the flow diagram that AI for early signal detection, how early signal can be detection with the help of AI. So if we see the first, the data collection, it likes adverse event, EHR and social media with the help of, then AI analytics with the help of AI, because clinical data is very huge and big data. It cannot be visualized and read by the human in a very less time. But our market is very potential and highly growing, so we need the results in a very quick time. So better option is to use the software, to implement the software and use that AI analytics. Then signal detection with the help of prioritization and validation. Then regulatory action. Once the signal has been detected, it should be go to the regulatory authorities, and then finally they should act on it, whatever the action it should be. Then feedback loop, that continuous monitoring should be done. Then this is the comparison between the traditional and the technology based. If you see the traditional oversight, if you can see the delayed signal detection, limited data sources, reactive monitoring and slow response. Whereas in the technology enabled oversight, if you see, it starts with the AI, it ends with the predictive risk monitoring. Predictive risk modeling is nothing but the simulation modeling. If you aware about the simulations, we can provide the in vivo, in silico models, and with the help of that, it can be done, and it will leads to the proactive safety management. Then there are a few more tabular format information where the AI for early signal detection, big data analytics and all have shows their key function, regulatory guidance, reference, because the reference of guidance is much more important before utilizing or implementing any concept in the regulatory affairs. Without guidance, without evidence, without any base, regulatory will not use any further point. So, and the last point is the impact on the drug safety oversight. So there are a few agencies which currently utilizing these things. If you see the EMA, they are using the Erato enhanced review of abstract with transformant model, and these three software, if you can see in the table, and the purpose and impact is also mentioned, that using the first one is used for the use for transforming based NLP to screen the literature adverse event signal, which saved the more than 800 hours for the pharmacovigilance review time in 2025. So if you see, this is the real case and the real live example which the AI drives us very fastly and we reach to our goal very fastly. Then there are COMs and WHO and FDA also using the AI which help us to drive the drug development into the market faster. Then patient-centered regulation. What actually the mean? Name itself indicates the regulation should mean in a way that helps the patient to get the safe medicines and shows the efficacy in an appropriate way. So if we see this diagram, so patient can report their adverse effects through the mobile app, which is currently very booming in the picture. Then regulatory can review that information from individuals' devices, and then risk assessment and decision happen, and then finally communication, and it leads to the feedback and trust between the patient, regulatory authorities, and the organizations. So there are few areas where it has been already happened and the process is going on, like focus areas, description, guidance references, and the impact. So these are the few regions where the program and case studies are actually given. Like for Africa region, community pharmacovigilance pilot program. These are the recently started program considering the patient safety into the market. Then Europe, America, Asian, and global. Then what would be the future outlook for this? That what should be the action of the future which can be drive patient safety in the better way? So the adaptive regulation of emerging therapies. So we should not stuck into only the pharma, because we are studying the pharmacy only for the medicine, but beyond that, the monoclonal antibodies, vaccines, enzyme, protein, peptides, and lots of things, gene therapies also are there, that should also be considered as the emerging therapy, and that should be adaptive as a part of regulatory affairs. Then predictive model anticipating risk. So before publishing any article or before driving our molecule into the market, the predictive model should be there which anticipate the risk, and it should be done during the clinical phases of the molecule. Then seamless global safety ecosystem, AI-driven safety which help us for the real-time monitoring powered by the advanced analytics. Then inclusive regulation and unified standard. Unified standard, it's well-known concept. So global regulatory alignment through the WHO-led harmonization. Then these pictures tells you the future outlook in very quick way, that it should started with the patient, drive by the technology, and finally leads to the global harmonization. And it should be the AI driven so that it would be much more efficient. So we'll reach finally to the conclusion, what actually conclusion should be from, and what should be the call to action for the regulatory managers, regulatory affairs people, and the pharmacovigilance system. So the journey from traditional oversight is technology enabled, patient-centered, globally harmonized regulation is underway. Then the challenge now is not an innovation but the commitment, because developing medicine is not a much challengeable, because we have good scientists in our country, but ensuring the stakeholder act together to scale this worldwide is the biggest challenge. And then regulators commit to transparency, adapt harmonized standard, and embed the patient voices. Then industry's role will be implement the ethical AI. AI should not be the blunt, it should have the sense, should detect the signals properly. Then the safety data responsibility of the pharmacovigilance team and align with the WHO framework. Then academia and public health. So academic also plays an important role here. What that role should be? Drive research that bridges the technology with patient outcomes. So I just want to exaggerate this point here. Recently, it's a very big thing that many of academia are coming to the industry and industry is going to the academic. The best example recently I show is the Naipur Mohali. they have merged with the biggest agencies and the biopharma production hubs. They are developing the technology into the upper Mohali, and then they transfer their scale and technology to the industry. So this is the one type of collaboration can be happen and can be very meaningful for this patient safety. Then global network that scale WHO-led convergence initiative to ensure the equitable access worldwide. So there are a few things like in this tabular manner, the technology, patients and global harmonization. So if we see the technology, like AI tools which invest in interpolate ethical and AI system across the regions, patient reporting portals are there. Lots of health agencies already established their portals, so patient directly log in their portals and report their adverse effect, whatever they are, and expand the patient representation feedback globally. Then global harmonization, we see the WHO working well on this point and strengthening the convergence capacity building. So these are the few references which can be utilized to dig further to have a better pharmacovigilance and the regulatory interlinkage for the patient safety. And that's it from my end. Thank you. Thanks for your attention. So anybody, if have any question, the floor is yours. You can raise any. Question.
Thanks, Shubham, for highlighting such a nice points in your presentation. My question is related to that. So, that these young B.Pharm students or M.Pharm students, if they want to enter into the pharmaceutical industry, now we know that today's is the AI era. So, before that industry, they are going to take the student only based on the basis of their knowledge and interview. But right now, their scenarios complete change, and they are going to ask what your skill in AI, so that they don't replace the existing staff with the AI. So, what your opinion in that particular thing?
Sir. Sir.
Yeah. Thank you, sir, for this question. I think it makes sense to me as well, considering the generation coming to the industry level. So definitely AI is much impactful factor, which agency expect from the upcoming newcomers. So my suggestion and my personal experience is that if students during their academics year learn some software, there are lots of software into the market related with the regulatory affairs and the pharmacovigilance system as well. They have their certification courses and you can say the fees, but yes, these courses, if they do, then definitely they accepted and adapted into the industry. Recently, a few of my juniors adapted by some well-known MNCs, and what was their benefit? They did some good AI software trainings. Because of that, they were able to manage the huge data in the small time and deliver the result in the efficient way. So that agency, the health agency as well as the regulatory organization were impressed, because they were seeing that lots of time can be saved by that people, so that they can hire them and they saved and they should not replaced with any AI. So my suggestions for the students would be only one: grow and learn. So currently there are lots of softwares and trainings are provided by the FDA, Europe Medicine Agency and the WHO as well. You should visit their sites. You should learn that software. Few of them are free. You can just visit them and you can just learn them, just log in. And few of them need some fees, but it's a minimal fees. It should not be the higher one. So I would suggest students during your graduation, not in the first or second year, but when you are in third or fourth year, visit such a sites, visit through the WHO, EMA, FDA sites, learn that thing, use and utilize the webinars. In webinars also they provided you the material which can be utilized to develop your AI trainings. So that would be the probable solution for based on which students can easily imbibe into the industry, and industry will also accept them. Because we know industry also needs the experience. They don't hire or they bother to hire the freshers one because they don't know, they don't need to utilize or invest their resources to learn or to train you, but they need the train material. But where you can get trained? So you can get trained into such environment like third or fourth year. You can attend the webinars, conferences scheduled or done by the agency, so that you can be on a track to the various organizations.
Thank you, Shubham Chinchulkar sir, for the enlightening presentation, for sharing your groundbreaking research on and helping us navigate the future of pharmaceutical science. Your dedication to advancing patient care and innovation is deeply appreciated. That was truly a fantastic session. Your note over EMA, MHRA, US FDA compliance, explanation about global harmonization, leveraging technology, patient-centered regulation will truly helpful for our budding pharma students. Thank you, sir. Now moving towards the appreciation ceremony, to express our deepest gratitude for your invaluable presence and insight, we would like to present a small token of appreciation to esteemed guests. May I request Ms. Kajal Bondre ma'am to please grace the stage to do honor and present token of love and appreciation to Mr. Shubham Chinchulkar sir on behalf of Adarsh Institute of Pharmacy. Thank you, Kajal ma'am. Thank you for joining us and for your valuable presence. We are truly honored. Thank you, sir. Now, for next proceeding, I would request my fellow colleague, Ms. Kajal Bondre ma'am, to grace the stage. Thank you all for giving me this opportunity. Thank you.
Good morning, respected Chief Guest, Honorable Principal Sir, distinguished dignitaries, faculty members, and my dear students. I am Kajal Bondre, Assistant Professor, Adarsh Institute of Pharmacy, Nagpur. It is my great honor to welcome you all to the National Conference on Integration of Pharmacovigilance and Regulatory Affairs for Enhanced Drug Safety. Today we are privileged to have with us Dr. Darshan Telange Sir, an eminent academician and researcher in the field of pharmaceutical sciences. He is currently serving as Associate Professor and HOD of Pharmaceutical Regulatory Affairs, as well as Dean Research at Datta Megi College of Pharmacy, Wardha. Dr. Telange Sir holds a PhD in Pharmaceutical Sciences and brings over 12 years of rich academic and industrial experience, including his role as an IPQA officer in the pharmaceutical industry. Sir has made remarkable contribution to research with more than 50 publications in reputed journals and a strong citation record. He has successfully guided PhD and MPharm scholars and has been actively involved in research administration and quality initiatives. His achievements also include multiple patents, copyrights, and authorship of books and book chapters with international publishers. His core research interests lie in phytosomes, nanocarriers, solid-state characterization, and quality by design approaches. We are truly honored to have a distinguished personality among us today. Now I request Ms. Abhisha Rokde ma'am to kindly felicitate our esteemed chief guest, Dr. Darshan Telange sir. Thank you, sir. I would like to request sir to kindly deliver his expert talk on topic regulatory and pharmacovigilance challenges in nano-enabled drug delivery system. Please.
Okay. So thank you, ma'am, for such a nice introduction.
तुम्हाला पाहण्यासाठी वगैरे नेक्स्ट स्लाइड.
Okay, so myself, Dr. Darshan Telenge, that working as an associate professor in the Datta Mege College of Pharmacy. Now, my topic of presentations that include the regulatory and pharmacovigilance challenges in a nano-enabled drug delivery system. But before that, entering into the details, let me tell you the overview of this particular topic. And this topic is how it is aligned with your conference theme. So we know that, so when we considering the nano-enabled drug delivery system, so this topic is something new for the UG, but with those UG, when it is entered into the PG and PhD, they're going to know that what exactly the nano-enabled drug delivery system. So it is nothing but the nanotechnology-based drug delivery. So we know that all. So this is the era of AI along with the nanotechnologies. So we are going to work on the nanotechnology. We know that this nanotechnology drug delivery system will offer the targeted drug delivery system as well as the therapeutic efficiency to the patient also. But when we are developing such type of the drug delivery system, we received some challenges. And that challenges that include the complexity of that particular formulations, okay, behavior of that particular formulation, physicochemical characterization, and when this formulation is entered into the body, so they are going to show the some different mechanism. So these are the mechanism by which it creates some challenges. So that is the first one, which involves in the regulatory. But when we are comes under the umbrella of the pharmacovigilance, which is a vast one. So based on that, so when we are using the pharmacovigilance, because pharmacovigilance is also important, we cannot think about that about one. So when we are considering the pharmacovigilance, so this pharmacovigilance actually tracking the adverse event of that particular formulations, okay. Thereafter the again physiological behavior of that particular formulation and the lacking of the reporting according to the regulatory framework. So these are the again challenges happening in this particular pharmaceutical industry. So for that particular creating the challenges or fulfilling the challenges, we need to integration of these two, that is regulatory as well as the pharmacovigilance. So when we are integrating regulatory as well as pharmacovigilance science into the nano-enabled drug delivery system, then definitely we can implement such type of the challenges as well as use this type of nano-enabled drug delivery system to the patient effectively. Okay, so before that, let me enter you into the nanotechnology. So we know that there are multiple definitions of the nanotechnology. You can Google it, you can AI. So there are multiple number of the definition you can get. So first, we're going to see that. So that nanotechnology is nothing but a drug delivery system that actually gives the particle size range within the within the 1 to 100 nanometer, and that actually provide the safety, stability, and targeted delivery to that particular formulation. Thereafter, another one that is nothing but it is a engineering formulation system that manipulating the pharmaceutical excipient. According to the manufacturing, according to the procedure, we can engineer that particular materials, and based on that, it will create the nanocarrier, and that nanocarrier will actually provide the controlled release, then a sustained release within the biological system. Thereafter, another definition is nothing but it is the use of the nanostructured carriers. So that nanostructured carriers means involves the nanoparticles, that involves the liposomes, that involves the phytosomes, that involve the SLN, NLCs, thereafter the neosomes, phytosomes, and multiple number. So there are main purpose is nothing but just to increase the solubility, okay, just increase the bioavailability, and finally the pharmacokinetic performance of that drug. Okay, so based on that, we're going to cover now these are the different examples of the nanotechnology based drug delivery system. So first, if you are going to include the nanoscale drug delivery system. So this nanoscale drug delivery system, it is having this particular nanoparticle. So this nanoparticle, if I look at this nanoparticle, then this nanoparticle shows that it is a one of the polymer, and that polymer is having the sheath, okay? Then there is having the drug molecules, and within that drug molecules, that having the nanoparticle around the 1 to 100 nanometers, and this nanoparticle going to attack on that particular disease state according to the target. Thereafter, so this is the main role. What is the main role of this particular drug delivery is nothing but that it improves the stability of that particular formulation, then the solubility and the targeted delivery. Thereafter, the another one, that is a controlled and targeted delivery. Now, why we are going with the different types of the drug delivery? So there are nothing but so these are the release nanocarriers. So these release nanocarrier could be the liposomes, could be the targeted liposomes to the functionalized liposomes, okay? So these liposomes, again, it is made up of nothing but the multiple number of the phospholipid, thereafter the different types of the copolymer. So based on that, it will create the polymer matrix to the outermost shell, and then there is having the drug reservoir, and the drug reservoir, maximum times we can going to select the BCS class 2 or BCS class 4, that having the low solubility and high permeability. So because of this particular behavior, or we are providing this particular behavior, it will provide the controlled release, means we can modulate that release more than 24 hours, okay? So for that, it can reach to the target cell, and that target cell will actually recover from that particular disease. And this again, the role is again controlled, sustained and site-specific drug release carrier. Thereafter, the another nanocarrier types in the drug delivery. So these include the various nanostructure. That various nanostructure include the liposomes, which is a well-known type of the drug delivery, then the polymeric nanoparticles, then the solid lipid nanoparticles, and the dendrimers. So these are the advanced drug delivery carriers. So their main role also the bioavailability enhancement and the pharmacokinetics. Bioavailability, everyone knows what is exactly bioavailability, which is nothing but the increase the rate and extent of that particular drug within the blood circulation, so that it can maintain to the maximum period of time and it reaches to the target without any leakage from that particular formulations, okay? Thereafter, the targeted drug delivery mechanisms, especially we can design it for targeting the cancer. There are multiple types of the cancer. There is a breast cancer, liver cancer, lung cancer, brain cancer. So for how we can going to treat that particular drug delivery formulation. So for that, we can going to modify the simple conventional formulation by adding the ligand molecule, okay? So by adding the ligand, what exactly happens? So it gives the targeting to that particular molecule, and this targeting will going to attach to the overexpressed receptor which is found on the any type of the cancer, okay? So based on that, we can again provide the precision targeting. It gives the precision targeting, thereafter the improved therapeutic outcomes, and then reduce systemic toxicity. And the last one, that is overcoming the biological barrier. Especially it is used for the transdermal drug delivery system. We know that there are conventional formulation, that is a cream, gel, then the lotion also. But this type of the conventional formulation doesn't achieve that much type of the therapeutic efficacy. So for that, we can also going with the advanced formulation, and that advanced formulation should break this barrier or overcome this barrier, that is a stratum corneum, which is the one of the toughest barrier of the skin, okay? So this overcome should be required, and for that we require the advanced based drug delivery system, and that will gives the efficient transportation of the drug through this epidermis, endodermis, and then into the blood circulation, and then it going to provide the autoimmune diseases like psoriasis type of the drug delivery, okay? Thereafter, the importance. What exactly the importance of this drug delivery? Importance, we said the significance. Why there is a important, why it is a significance? Because we know that first it is actually going to improve the solubility of the BCS class 2 drug or BCS class 4 drug. We know that BCS class 2 it is a low soluble and high permeable, but BCS class 4 it is a low soluble and low permeable. So for achieving that particular parameter, we need to go with the advanced drug delivery. So this advanced drug delivery, what exactly does? It does to reduce the particle size of that drug, increase the surface area, and because of these two tacklement, it increases the solubility, dissolution, bioavailability, and finally the therapeutic efficacy. Thereafter, they improve the oral and systemic bioavailability. Formulation not only improving the solubility doesn't make any sense. So for that, you need to increase the solubility, but along with the solubility, bioavailability should also goes to increase, means the drug should enter into the blood and maintain in the blood for longer period of time, then it can achieve the bioavailability. Thereafter, the targeted drug delivery to specific cells, as I discussed in the last slide, that we required the functionalized nanocarrier, targeted nanocarrier. So for that, we can achieve just by designing of this nanocarrier by adding specific ligand molecule. For example, that is a folic acid. For example, that is a hyaluronic acid. So these are the two ligand molecule which is specially occurred or expressed on the type of the cancer cells. Example like a liver cancer cell, lung cancer cell, brain cancer cell, and breast cancer. So this type of the cancer, when it is in a pathogenic condition, they actually develop some signals. They develop actually some protein molecule, and that protein molecule we can recognize as a ligand, okay, as a receptor. So this receptor going to receive this particular ligand. So there is an interaction takes place, drug or ligand and the receptor interaction takes place, and this interaction will establish the targeting of the nanocarrier to that particular formulation, to that particular cell. So that we can achieve with the help of surface modification that will achieve the active targeting as well as the passive targeting. Thereafter, the controlled and sustained release, we know that again the development of the nano drug delivery system not only achieve the bioavailability, but it should achieve the controlled and the sustained release when it is having the acute condition, when it is having the emergency condition. So for that emergency condition, we can avoid the frequent dosing, and for that avoiding frequent dosing, we can go with the controlled release. So instead of patient get the frequent dosing, he can get the once a time, and he can get the control for maximum period of time. Thereafter, the enhanced permeability and retention effect, which is again an important part of the advanced drug, nano-enabled drug delivery system. So this is the part of the passive targeting. So when we need to go with the simple formulation, conventional formulation, but few modification, not advanced modification. If you are going with the few modification, then in that condition, what will happen? That particular formulation going to attack on the cancer cell, and it will provide the enhanced permeation and retention effect, directly goes to that particular cells, and it will give the targeting effect. Now the reduction in dose related toxicity and side effects. So based on this particular point, we can achieve the last point, that is by advanced drug delivery system, we can achieve the target. By achieving the target, definitely we can reduce the off-site target, and because of that off-site target, we reduce the systemic toxicity and achieve the maximum therapeutic efficacy. Now the rationale. Rationale means nothing but the need of the study. Why there is a need? ये क्यों करना चाहते हैं? So for that, again we are having the, because of having some limitations. Limitations means some weakness, some unaddressed questions of this conventional formulation. Conventional formulation, that is solution, elixir, simple gel. So this particular formulation, it is in the market, no doubt. It is a high acceptability by the patient, but this still formulation having some challenges, and that challenges is having the solubility issues, having the below bioavailability issues, and non-specific distribution within that particular organs of the body. And that's why the patient need to take that formulation in a frequent manner, in a frequent dosing. Okay, so that is the first limitation. Second one, inadequate targeting and high systemic toxicity. So because of this conventional formulation and because of the high dose of taking, because of that, what will happen? It gives the inadequate targeting, and because of that inadequate targeting, it increases the systemic toxicity to that particular patient. Thereafter, need for improved delivery of complex and poorly soluble drug. So we know that according to the synthetic chemistry, or according to the synthesis, that is a high throughput screening technology has been used for the development of the multiple number of the new chemical entity and new biological entity. But when it is in the screening process, but when it is in the clinical trials, maximum number of the new chemical entity and new biological entity have been failed. Why? Because of having the solubility issues, because of having the permeability issue, and that's why they categorized into the BCS class. BCS class 1, BCS class 2, BCS class 3, and BCS class 4. Okay, and maximum they are again categorized within the class 2 and class 4. Okay, so therefore, now advantages of the nano-enabled system. So these advantages, so when we are converting these parameters into the advanced one, what we achieved the, again, now we achieve the, this enhance, we can achieve the formulation, but this formulation again will going to increase the solubility that could not be possible to achieve in the conventional drug delivery system. Thereafter, the stability. Stability means Stability means we need to maintain the drug within that carrier for the maximum period of time without any leakage. क्योंकि maximum formulations में क्या होता है कि when we are going into the stability, maximum formulation they get failed to pass the stability according to the ICH guidelines. Okay, I think Shubham knows very well. So for that we need to more and more work on the stabilities also. So therefore, again the permeability. Permeability means not only on the transdermal drug delivery system, but again when it is go with the oral route of administration, so this has to be passed through the plasma membrane, and that plasma membrane, it is again the lipidic in nature, which is not possible to get past the aqueous based drug delivery system. So these are the major challenges, and it has to be overcome with the help of these advantages of the nano enabled drug delivery system. Now the targeted and efficient drug delivery. So this nano system, again it will give the passive targeting benefit and active targeting benefit. Passive targeting means without any decoration to that conventional drug delivery system, and active targeting means decorating the drug delivery by adding the ligand molecule to that formulations. Okay. Thereafter, the growing regulatory acceptance and approvals of the nanomedicine, as we have done our work. But what about the regulatory approvals? What about the pharmacological things? So for approving that formulation, and once it is entered into the market, it requires multiple number of the challenges, and that we need to tackle in these particular presentations. Okay. So now these rationales. So these are the diagram where we are giving the limitation part and also the advantages part. So consider this is the nano-enabled drug delivery system. So that include the nanoparticles, that include the liposomes, nanospheres, dendrimers, and phytosomes. Okay. So these are the limitations. So first include the, as we know that having the more acceptable pharmaceutical doses from in today's scenario, tablet capsules, we know that. But still tablet capsule, it is in the market and it is accepted by the patient. But still, it is having the problem that is having the low soluble. Now why? Because that tablet contains the BCS class 2 drug and BCS class 4 drug that doesn't giving that much efficient therapeutic efficacy. And it could be only possible to converting into the advanced drug delivery system. Okay. The second one, that is low and variable bioavailability due to the first pass metabolism. So when we are taking the drug, taking the formulation by oral route of administration, the most of the chances going to the protein binding, and second one with the first pass metabolism. And because of these two major challenges, less amount of the drug reaches to the systemic circulation. Okay. Thereafter, the non-specific distribution. So when we are targeting, suppose if we are having the conventional, but this conventional drug delivery system doesn't having the ability to get entrapped within that particular specific tissues. Okay. Thereafter, the need for higher doses and frequent administration. So because of this time, the patient need to take the same medications again and again for the maximum period of time till to achieve its therapeutic level within the blood concentration. Okay. And then after, the instability and rapid degradation of the drugs in biological fluids. Okay, because of the some excipient which is not able to get maintained within the biological fluid. So because of that, what will happen? They get degraded. Okay, so these are the limitations and these are the advantages of the nanoable. If you are converting these into the nano-enabled drug delivery system, so we can receive the multiple number of the advantages. So improves the solubility, thereafter the absorption and bioavailability, thereafter the enable passive and active targeting, thereafter the provide controlled and sustained drug release, and thereafter therapeutic efficacy and improves the stability. Now there are unique characteristics impacting the safety. As we have discussed, these are the formulation where we can achieve the maximum number of benefits. But out of these benefit, which unique characteristics we required in the formulation so that it can provide the safety to the patient. Okay, so that include the first and most important, that is particle size and surface area. Because these are the formulation in which once the particle size, it is in a appropriate size or physicochemical characteristics, definitely it is entered into the biological media, then it is entered into the biological membrane, and then it is entered into the blood. Okay, so for that we require the particle size and its surface area to the inverse proportional to each other, because if the particle size is lower, surface area will be more. Okay. Thereafter, the surface charge and the functionalization. I think in the third sem, maximum time we have studied the subject pharmaceutical engineering, and in that the second chapter, that is size reduction. I think all of you have read this particular topic, size reduction. Okay, so within that size reduction, what will happen? We are reducing the size reduction for what purpose? Just to reduce the particle size to get the uniformity. And because of that uniformity, what we are achieving? We are achieving the uniform size and shape, and that uniform size and shape, it will produce the small particle size and increase the surface area, and that gives the high solubility, dissolution and permeability. But during this reduction, what will happen? Maximum time the surface charges are developed to the surface of that particular particles. Might be the positive charge, might be the negative charge. So this may actually create some collision, and because of that collision, again further reduction going to take place. So that could not be possible to get handled. So for that, we require the surface charge, but that surface charge should be positive or negative. Okay. Thereafter, the functionalization we can also achieve. Functionalization is nothing but the addition of ligand molecule to the surface. Advanced formulation. Okay. Thereafter, the biodistribution and the accumulation. Biodistribution and accumulation is nothing but the nanoparticle that exists distinct biodistribution pattern depending on the size, shape and surface properties which directly affect the safety and efficacy. So once these particular particle size of the formulation, if it is enters to the desired size and shape within that particular organ or within that particular tissue and distributed over there for maximum period of time, definitely it gives the maximum therapeutic efficacy. Thereafter, the interaction with the biological system, because when we receive the therapeutic response, we cannot measure it, we just experience it. But how it is possible? It is just possible because of having the interaction between the formulation and our biological system in the form of drug receptor interaction. So this drug receptor interaction, it could be possible just with the help of the designing of the drug delivery, where the nanoparticle interact dynamically with the proteins molecule, with the receptor molecule, biological fluids, and forming a protein corona, and thereafter which was alters nothing but the identity of that formulation and also the targeting ability, and it will create the safety profile further. So the unique characteristics of the formulation. Now, so when we considering the particle size and the surface area, so there are now, so there are multiple number of particle size. First it is a 10 nanometer, thereafter the 50, thereafter the 100, thereafter 200, and thereafter 500 nanometer. There are multiple number of the nanometer size, and these are all the small particle size. But out of these, which one is more suitable? We doesn't require the extreme lower, and we doesn't require the extreme large. We require the medium, that is 100 nanometer. Why? Because this gives the better tissue penetration and cross the biological barriers very easily, and that's why it gives the better therapeutic efficacy. Thereafter, the surface charge and the functionalization. As I discussed, we don't require the positive charge, we don't require the negative, but we require neutral one, positive as well as the some negative, because it gives the understanding of the biological behavior of that particular tissue, and according to that, the formulation will behave within that particular formulation, within that particular biological fluids. Thereafter, the biodistribution and the accumulation. According to the designing of the drug delivery, maximum times the drug may accumulate within the liver, then the spleen, then the tumor, and then the bone marrow. So because of this distribution and accumulation, the particle may interact with that particular tissue fluid or tissue proteins, and it can going to produce that particular targeting effect. Thereafter, the interaction with the biological systems. So when we are having this particular formulation and when it is entered into the biological fluid is nothing but the intestinal fluid. So within that intestinal fluid, we know that there are multiple number of the ions are present, various types of the proteins are present. So because of this, what will happen? They multiple times going to interact with that particular formulation, and it will create the size of that or increase the size of this formulation. And because of this, what will happen? It will increase the size, and that's why sometimes it going to interact with the tissue, or sometimes it doesn't make interaction with the tissue. So therefore, it again going to create the challenges for implementing or for increasing the therapeutic efficacy of that particular formulation. Now, the safety concerns in the nanomedicine. These are the all things that we required. But out of these, what are the safety concerns? Because of that, we can put into the regulatory agencies. So these regulatory agencies can determine that particular concerns, and according to that, give the approval into the market of this particular formulation. So the safety concerns, first, they're going to notice the cytotoxicity and oxidative stress.
Okay, because most of the formulation, they're going to exaggerate or they're going to stimulate the cells, and because of that, they're going to produce the reactive oxygen species. And in reactive oxygen species, there are multiple number of the radicals. And these radicals going to further affect the genetic media or genetic cells of our body, and because of that, the further degradation going to be possible. So example, that is silver nanoparticle, then cationic liposomes, thereafter the metal oxide nanoparticles. They actually inducing the stress by developing the reactive oxygen species, and therefore it going to create the cytotoxicity. So because of this, the regulatory affairs, they going to notice that one and may going to reject the particular review. Thereafter the second one, that is the immunogenicity. So in the immunogenicity, the nanoparticle activate the immune system, that is they may going to react with the dendritic cells, may going to react the lymphocytes. So because of this, what will happen? So this actually create or initiate the immunogenic reaction, and because of that immunogenic reaction, some autoimmune diseases generation may going to be possible, that is the psoriasis and again type of the skin reactions. So therefore, for that, we require the pegylation. So this pegylation will actually reduce that particular immunogenicity, that direct contact with the drug and the immune system. Then the organ accumulation. So in the organ accumulation, so these nanoparticles, as we discussed, the particle size should not be too much less and should not be much more. Within the desired particle size, so what will happen? So this desired particle size doesn't get recognized by the reticuloendothelial system, which is a part of immune system. क्योंकि अगर ये recognize हो गया, then what will happen? It will be engulfed and removed from the body. So it will now be utilized by the body for its therapeutic purpose. So for that, we require the desired size of the formulation so that it doesn't get recognized by the reticuloendothelial system and maintain within the blood for maximum period of time and goes to the target site and provide the therapeutic effect. Thereafter, the nanobio interaction. So this nanobio interaction, again the nanoparticle in the biological fluid, it going to make interaction with the pH, it going to interaction with the enzymes, it going to interaction with the proteins, and that's why it creates the multiple number of the targeting ligands into the surface, and according to that, it may going to interact with the tissues. Now the regulatory landscape of the nanomedicine. So if we are doing such type of the things, then which particular regulatory pathways we require, which regulatory things we require. So first, complexity in the regulatory nanomedicine. If you are developing the nanomedicine, which is very good, no doubt, paper publication is done, patent is also granted, no doubt. But what about the complexity? How we are going to solve this complexity? And regulatory agency don't want that one. They want very clear and simple formulation. That's why the tablets, capsule, they're approved very easily. Why? Because they don't have any complex thing. But when we are going into the nano drug delivery system, it is having the multiple number of these types of formulation, multiple number of the excipients involvement, multiple number of the characterization. So because of that, the regulatory agencies, they don't bother about the regularization of this formulation. But we need to get it clear. But how? Okay. So therefore, evaluation challenges. Why? Because of having the size issues, because of having the surface characteristics problem, then having the behavior in the biological system following upon oral administration, transdermal administration, or parenteral administration. So the lack of universal definition. Each and every country having their own regulatory pathways. There is no any harmonization, and because of there no harmonization, there is no any common platform for getting approval of this particular formulation, and that's why again it receives the maximum number of challenges. Thereafter, need for product specific evaluation. Product specific means, suppose if you are having a cancer based any nano formulation, then this nano formulation having one particular channel, having one particular pathways. Okay, so because of not having these particular pathways, we are going to require the product specific evaluations and again evolving regulatory guidelines. But because of these particular problems, the regulatory agencies, they are updating themselves, no doubt. But for updation, they require some time, and because of the time, it takes maximum number of the formulation to get pending for the approval. Now there's these key regulatory challenges. So includes again the same thing that I discussed. Doesn't work. Now the quality and characterization requirement. Now which quality and characterization things are required? So first one, that is the particle size distribution. That is one of the most important, because its negligence will affect the final approval. Thereafter, the zeta potential. Anyone knows what is zeta potential? Anyone knows? Zeta potential is one of the indicator which decide the physical stability of the formulation, and it should be within the range of plus and minus 30 millivolt. Okay. Thereafter, the drug loading and entrapment efficiency. As I told you that stability, okay, the nano formulation gives the or offering the maximum stability, but on what basis it gives the stability? It gives the stability on the basis of this entrapment efficiency. If the nano formulation gives the maximum entrapment within that particular formulation, then definitely that formulation is stable, no doubt. But this formulation, while traveling from administration to the target site, it should be maintained within the same thing. There should be no any leakage. If there is no any leakage, then we can say that it is a stable. But if there is a leakage takes place, then the formulation may get destabilized. Okay. Thereafter, the release kinetics, again, the modulation we required, and for that modulation, we required the, again, decoration of this drug delivery with the advanced material, with the advanced technology, by which we can convert the immediate release into the sustained and controlled release formulation. Then the surface morphology, again, surface morphology doesn't make that much requirement. So the collectively, that ensuring the consistent performance, so all this formulation performance, then safety and reproducibility of that particular formulation. Now again, the pharmacokinetic challenges. ये सब तो रहेगा, but apart from that, pharmacokinetic challenges also create the big difference, and that difference is ADME. What is ADME? Everyone knows? It's absorption, distribution, metabolism, and excretion. Each and every drug profile having the different ADME. So absorption, distribution, metabolism, and excretion is actually affected to nano formulation to formulation. So for that, we need to get it maintained. Thereafter, the non-linear kinetics. Non-linear means it should not be a straight line one. It should be the like a valley type. So this valley type, what it indicates? It indicates that at the time of administration, the absorption is less. But once it is in the middle, it gives the maximum absorption, and then it is removed from the body. Thereafter, the enhanced permeability, again retention effect. Now just look at these two diagrams. So this is the clear image of the normal tissue. So within that normal tissue, there will be no any deposition of the nano formulation. But when it is in a leaky form, when it is in a pathogenic condition, the nano formulation going to deposit over there, and they actually provide the therapeutic effect, and that's why it is called as enhanced permeation and retention effect. That is a passive targeting. Thereafter, the difficulty in predicting the in vivo behavior. We don't know how the formulation will going to behave once it is administered. So for that, we also going to predict with the help of the AI or in type of the particular software, so that we can determine the exact of the nano formulation, so that we can provide that evidence to the regulatory agency that it gives the same thing as we expected, and that's why it gives the key evidence for its approval also. Now the pharmacovigilance. Till now we have considered about the regulatory affairs. But what about the pharmacovigilance? So this pharmacovigilance will going to work once it is approved by regulatory agency, no doubt. It is approved as it is approved by the tablet capsule. The same thing is approved by the regulatory agency for the nano formulation. It is in the market. But what happens after that? Either this formulation working properly, either this formulation gives the better benefit to the patient, yes or no. So for that, we require the post-marketing surveillance. Continuous surveillance we require, so that we can give the latest update, and that latest update we can submit to the manufacturers, and that manufacturers can submit to the regulatory agency, so that maintain the communication for the particular formulation. Thereafter, detecting the rare long-term adverse effects. Suppose at a first year of that particular regulatory approval of the formulation, it is used by the patient, but the first year doesn't make any problem to the patient. But what about the second year? What about the third, and what about the final? So that particular thing should be recorded for the long term adverse effect, and then the need for personalized monitoring system. Personalized, that requires the tailored one. Tailored one means we can modify the pharmacovigilance system according to the we required, not according to the system. Okay, and thereafter so these are the one example that I've quoted over here. So that example is, so the liposomes drug formulation have shown infusion related reaction. Infusion related, that is nothing but the hypersensitive reactions, delayed toxicity during the post marketing surveillance, highlighting the need for targeted safety monitoring. So it doesn't guarantee that once the formulation it is regulated by the regulatory agency and is entered into the market, doesn't guarantee that it will give the therapeutic efficacy. So for that we require the continuous monitoring. Now the challenges in the pharmacovigilance. What particular challenges we are having? Again, the lack of specific reporting system for the nanomedicine. There is no any regulatory pathways. There is no any regulatory pharmacovigilance systems. Till now it is updated in the system so that we can give the perfect reporting. Thereafter, difficulty in identifying non-specific adverse effect. There is no any system over there. So how we can identify? It is if it is identified, but that one it is a very vast one. There is no any specific. So for that we require the specific identification of the adverse effect. Now the limited real world data. Whatever the data we are having or whatever the data we are collecting, it is only the generalized one, not a specific one. So for collecting that specific, we require the strengthening to that pharmacovigilance system and the traceability issues. Traceability issues means, suppose if you are going to identify that this issue or this reaction or this adverse effect is given by this particular nanoformulation, but still we doesn't get identified because of not having the traceability mechanism. Okay. So how we can manage that particular strategies? Okay. So for that, preclinical toxicity studies before giving the approval to them for that particular formulation for entering into market, we need to go with the preliminary toxicity studies. Preliminary toxicities, that is acute toxicity studies as well as chronic toxicity studies we should require. Okay. Thereafter, the life cycle approach to the safety, that undergoing surveillance, it should be again maintained in the surveillance for the maximum period of time for the maximum years so that this formulation get safe for the patients. Thereafter, the risk-based assessment. So the reduced cardiotoxicity of the liposomal doxorubicin, it should be again the risk of hand and foot syndrome. Most of the times, doxorubicin liposomal formulation we are preparing for reducing its cardiotoxicity, but apart from the cardiotoxicity, it also creates the another important disease, and that is nothing but the hand and foot syndrome. So the again signal detection and evaluation part. So the detection of infusion related hypersensitivity reaction in a nanoparticle based nanoformulation through the pharmacovigilance database should be required. So we need to maintain the pharmacological database. So this database, once it is entered, this database into that particular adverse reaction, they're going to identify very wisely. Okay. So now we are going to focus on the one case study. So this, whatever we have studied over here, so how we can going to implement in the one particular type of the case study, that is liposomal doxorubicin solution. So backbenchers, please stay silent. Okay. So now, so the first one, as we know that doxorubicin, it is one of the most resourceable drug for the use of anti-cancer. But apart from the anti-cancer, it also produces the cardiotoxicity. So how we can reduce that cardiotoxicity? So for reducing that cardiotoxicity, we are going to prepare the liposomal. But liposomal, it is a conventional. So we are giving the advanced one, that is pegylation. Pegylated liposome. So this pegylated liposome, what actually it does? It actually accumulates within the tumor tissue with the help of EPR. EPR means enhanced permeation and the retention effect. Then it reduces the exposure to the healthy heart tissues. Okay, so therefore what will happen? It will reduce the cardiotoxicity and it will increase the maximum accumulation within a cancer cells. Now the altered pharmacokinetics. Now see, simple liposomal formulation, now see, conventional liposomal formulation, it only gives the sufficient number of the therapeutic effect. But when we are converting into the advanced one, now see, its AUC and its T-half and its clearance will be maintained for the maximum time as compared to the normal one. So these gives the prolonged circulation. Thereafter, the reduced clearance and also increase the AUC. AUC, that is area under the curve. So that indicates the drug is maintained within the blood circulation for maximum period of time. Thereafter, the observe adverse effect, that is a hand-foot syndrome. So this hand-foot syndrome reported in some patient because of having the different types of the off-site targets. So the dose limitation, if you are maintaining the dose of that particular formulation for specific period of time, then definitely we can reduce this hand-foot syndrome. And the last one, that is regulatory consideration, bioequivalence challenges. Bioavailability and bioequivalence. Bioavailability we can maintain, but what about the bioequivalence? In liposomal and in advanced liposomal, we cannot make a we can say that we cannot similarize that this simple liposomal and its advanced liposomal will give the same bioequivalence. Doesn't never happens. If you are going for the liposomal, they need to go with the bioequivalence. But if you are converting into the advanced one, then still you go with the bioequivalence studies. Now the key learnings. So these are the problematic condition that we have observed, but we have tackled. Now what exactly we need to understand with this? So first one, nanocarrier changes the toxicity profile. Before that, it was a cardiac toxicity, but once it is converting into the nano formulation, we can remove the toxicity profile. Second one, need for the nano-specific safety evaluation. And third one, that is importance of the post-marketing surveillance. Now another case study is that is paclitaxel nanoparticles. So once the paclitaxel, anyone knows what is paclitaxel? Everyone? It is a plant-based taxol derivative, and it gives the anti-cancer effect, especially all types of the cancer. But when we are preparing into the simple formulation with the help of the chemophore and ethanol. So this chemophore and ethanol, it actually produces the toxicity. It actually produces the toxicity, and that toxicity will actually reduce the solubility and another important therapeutic benefit. But if you are going to prepare the paclitaxel nanoparticles without any use of solvent, that is simple with the help of green solvent-based technology. Green solvent-based technology. So with the help of this, what we can reduce? We can reduce the toxicity profile of this particular formulation. Now again, improve the safety compared to the formulation. So this taxol formulation, taxol-based nanoparticles, it actually increase the systemic toxicity, increase the neutropenia, and increase the greater pre-medications. But when we are converting into the nanoparticle, so what will happen? Systemic toxicity will be reduced. Now the less myelosuppression, and then a fewer medicine, supportive medications we require. There will be no any burden of the multiple number of the medication we need to take. Now the reduced hypersensitivity, and then the regulatory focus on the comparability. Okay. Then so the key learnings within that one, that is formulation impact safety the significantly. So with the help of just removing the solvent, it actually impact the more and more focusing on the safety one. Thereafter, the importance of the excipient elimination by removing the excipient, that is a chemophore, and replace with the green technology. Now again, we can remove the toxicity profile and regulatory emphasis on the clinical outcomes. So the future perspectives in the nanomedicine safety. So if you are integrating the pharmacovigilance and regulatory, what future prospect we can predict for the nano-enabled drug delivery system? So for that, development of the nano-specific guidelines, we can achieve that one. Then AI and big data in the pharmacovigilance, we can use the AI or tailored AI mechanism-based system so that we can track the adverse event effectively. Thereafter, the personalized nanomedicine safety evaluation. Instead of the regularized formulation, we can go with the personalized, that is a 3D printing-based formulation, 3D printing-based tablet capsule formulation. So this can again use the basic effects. Now the global harmonization regulations. Global harmonization regulations means we know that each and every country, they are developing different types of the complex formulations. So instead of regularizing in different countries, can regularize all the regulatory pathways into one particular platform, so that it will be giving the more and more simple regulatory process to the regulatory agency and clears the pending formulation for its approval. Okay, so that is the conclusion part. So this conclusion part, what actually understand on that basis. So first, the therapeutic potential it gives, no offers, no problems. But still, it having the challenges, and that challenges is include the characterization part, safety evaluation part, and the long-term toxicity study. Now, there will be required the integration of the regulatory pathways and pharmacovigilance integration. We need to combine these two and regularize in a common manner. Now the requirement of the robust framework. Framework means when we are going for the different countries according to their regulatory pathways, there are different formats. Is it so? There are different formats. So can we maintain the common format for all this? If you are maintaining the common format for all, then definitely there will be no any challenge, or we can reduce one of the challenges from the number of challenges. Okay. So this is the conclusion. Now the finally, so the acknowledgement. So thank you, sir, and management for this opportunity, for giving this particular time to give the presentations, and also organizing committee for the taking this initiative for the giving some knowledge to the UG student as well as the other students from the other colleges. And finally thank you. So the question and answers from your side are expecting. Okay. So any questions? Because I'm expecting the questions from your side. So if you are having any questions, then it's better way to make an interaction with all of you. But if you are not asking the questions, then it doesn't make any sense. So please ask the question, whatever the question related to the industry, related to the particular formulation, related to the regulatory, related to the pharmacovigilance, you can ask. And don't get hesitate that कि मैं question पूछूंगा तो इसको क्या लगेगा, उसको कैसा लगेगा. So don't, क्योंकि अगर आप अभी question नहीं पूछोगे तो कभी पूछ नहीं पाएंगे questions आप. So that's why I'm going to tell you this one. So if there is no questions, then I'm going to end up my other presentation. Thank you, sir.
Thank you, sir. Hello. Thank you, sir, for your valuable and informative session. Sir highlighted the key challenges in the pharmaceutical sector related to targeted drug delivery system. Thank you, sir, for simplifying the complex concept of targeted and nano drug delivery system. As a token of our gratitude and appreciation, I request Minal Raut Madam to kindly present a memento to Dr. Telangi, sir. Thank you, sir. Now I request Mrs. Rushika Jaiswal ma'am to kindly take over the proceeding for the next session.
Thank you, Kajal ma'am. Now to grace the stage, I would request Mr. Dayani sir from Prerna Clinical Solution to come over this dais. I request Professor to come over the stage and felicitate Mr. Dayani sir. Now I request Mr. Chetan Chaudhary sir to come over the stage. I request Rupesh Dudhe sir to come over the stage and felicitate Mr. Chetan Chaudhary sir, Assistant Professor, Maharashtra Institute of Pharmacy, Brahmapuri. Now I request Dayani sir to address the gathering.
Very good afternoon to all of you. We can discuss something till the presentation will be on the screen. आप जितने लोग बैठे हैं स्टूडेंट्स यहाँ पे सब बीफॉर्म वाले हैं, फाइनल ईयर, सेकेंड ईयर, ओके. सो सेकेंड ईयर के बाद थर्ड ईयर के बाद fourth year, like that, okay. So at this point of time, have you thought about your career? Hello, can you hear me? At this point of time, anybody, क्या आप अपने करियर के बारे में सोचा है कि फार्मेसी कंप्लीट करने के बाद हम क्या करियर बनाएंगे, या फिर कौन कौन से करियर ऑप्शंस अवेलेबल है, अल्टरनेटिव्स अवेलेबल्स आपके पास? शायद नहीं सोचा होगा, क्योंकि अभी तो मस्ती करने का टाइम है, 2 साल के बाद सोचेंगे, सही है? सो डेफिनेटली यू पीपल, कुछ लोगों ने सोचा होगा, कुछ ने नहीं सोचा होगा, बट जितने भी करियर ऑप्शंस अवेलेबल है आफ्टर कंप्लीशन ऑफ योर ग्रेजुएशन इन दिस फील्ड, आई एम गोइंग टू टेल यू और आई एम गोइंग टू गिव यू सम ब्रीफ आइडिया अबाउट वन ऑफ द करियर ऑप्शन अवेलेबल with you after completion of this graduation course. आप में से कितने ऐसे लोग हैं जिन्होंने लाइफ में कभी मेडिसिन नहीं लिया? एनीबडी? या आपके कोई भी पहचान में, एनीबडी फ्रॉम योर फैमिली, फ्रेंड्स, एनीबडी देयर जिसने कभी भी मेडिसिन नहीं लिया है? और क्या आपको लगता है कि दुनिया में कोई ऐसा होगा जिसने आज तक मेडिसिन नहीं लिया होगा? फ्रॉम द डे वन ऑफ बर्थ टिल द एंड ऑफ द डे, हर कोई, छोटे बच्चे से लेकर बड़े होने तक सभी लोग मेडिसिन ले रहे हैं. आज छोटा बच्चा जब बर्थ होता है तब से मेडिसिन लेना चालू करता है, और आदमी जब मरता है तब तक भी दवाइयाँ लेता रहता है. अलग अलग स्टेज पे अलग अलग दवाइयाँ खाते हैं. ये दवाइयाँ बनती कैसे हैं? फॉर एग्जाम्पल, नॉट अबाउट प्रोडक्शन के बारे में बात नहीं कर रहा हूँ. ये दवाइयाँ जो हम खाते हैं, हम पूरे मतलब कॉन्फिडेंस से खाते हैं कि ये हमें क्योर करेगी. सही बात है? तो इतना कॉन्फिडेंस आता कहाँ से है कि डॉक्टर ने लिख के दिया, फार्मेसी से हमने लिया और हमने डॉक्टर की सलाह के अनुसार उसे खा ला. लेकिन यही कोई अगर दूसरा व्यक्ति आपको आके बताता है कि आपकी बीमारी की ये दवाई आप इसे खाइए, तो आप खाएंगे? नहीं खाएंगे. क्यों नहीं खाएंगे? क्योंकि हमें नहीं पता ये सेफ है या नहीं, और ये सच में काम करेगी कि नहीं. तो दो बातें हैं, सेफ्टी और एफिकेसी. ये दो चीजों का कॉन्फिडेंस आता कहां से है? जब हम दवाई लेते हैं, ये दो चीजें होती है, सेफ्टी होनी चाहिए और इफेक्टिव होना चाहिए, नहीं तो दवाई खाने का कोई अर्थ है नहीं. करेक्ट है? तो ये कॉन्फिडेंस आता कहां से है कि ये जो हम मेडिसिन खा रहे हैं वो सेफ और इफेक्टिव है ही नहीं? ये कॉन्फिडेंस आता है, इसके पीछे कई सालों लंबे चले क्लिनिकल ट्रायल्स के ऊपर. ये जो क्लिनिकल ट्रायल्स, सीरीज ऑफ क्लिनिकल ट्रायल्स होते हैं, एक्सपेरिमेंट्स होते हैं, इसके बेसिस पे ही हम बता पाते हैं कि ये मॉलिक्यूल सेफ और इफेक्टिव है कि नहीं. और इसी फील्ड को हम कहते हैं क्लिनिकल रिसर्च. सो क्लिनिकल रिसर्च इज नथिंग बट अबाउट ड्रग डिस्कवरी. आई थिंक प्रेजेंटेशन के बदले कुछ और स्टार्ट हो गया है. एनीवेज, क्लिनिकल रिसर्च ऐसी फील्ड है जिसमें हम बात करते हैं ड्रग डिस्कवरी के बदले. ड्रग डिस्कवरी मतलब क्या, कोई बता सकता है? मतलब आप लोगों को जो भी लेमन लैंग्वेज में आता हो, व्हाट डू यू मीन बाय ड्रग डिस्कवरी? ड्रग डिस्कवरी मतलब क्या, बताइए. थोड़ा सा कॉन्फिडेंस लाइए. गलत हुआ तो भी कोई फर्क नहीं पड़ता है. जैसे किसी के दिमाग में आ रहा होगा नया ड्रग बनाना, नया मॉलिक्यूल बनाना, लाइक दैट. एक साइंटिस्ट लैब में जब एक नया मॉलिक्यूल बनाते हैं तो वो मॉलिक्यूल के ऊपर में कई सीरीज ऑफ एक्सपेरिमेंट्स होते हैं, कई सीरीज ऑफ क्लिनिकल ट्रायल्स होते हैं, जिसके बाद वो मॉलिक्यूल फाइनली मार्केट में आता है, फार्मेसी शॉप में बिकता है, जब उसकी सेफ्टी और एफिकेसी कंफर्म हो जाती है. दैट इज व्हाट द ड्रग डिस्कवरी. पर ये सुनने में जितना आसान लगा है उतना आसान है नहीं. इसके पीछे कितने ही साल लग जाते हैं, कितने ही पैसे लग जाते हैं, कितने साइंटिस्ट डॉक्टर्स की मेहनत लगती है. तो ये वन ऑफ द करियर ऑप्शन है आप लोगों के लिए जो ये फार्मेसी बैकग्राउंड कंप्लीट या ग्रेजुएशन या पोस्ट ग्रेजुएशन कंप्लीट करने के बाद इसमें फील्ड में जा सकते हैं. तो आप लोगों के लिए क्या जॉब अपॉर्चुनिटीज रहती है इस पर्टिकुलर क्लिनिकल रिसर्च की फील्ड में, हम उस बारे में थोड़ी सी बात करेंगे. क्लिनिकल रिसर्च के बेसिकली 6 प्रकार के फील्ड होते हैं, जिसमें एक फील्ड होता है क्लिनिकल रिसर्च ऑपरेशन, एक होता है क्लिनिकल डेटा मैनेजमेंट, एक अभी अभी हमने रिसेंटली सुना क्लिनिकल फार्माकोविजिलेंस, एक होता है रेगुलेटरी अफेयर्स, मेडिकल राइटिंग और बायोस्टैटिस्टिशियन. इसमें हम बात करेंगे ज्यादातर क्लिनिकल ऑपरेशंस पे, क्योंकि यही बहुत ज्यादा कॉम्प्लिकेटेड और टाइम कंज्यूमिंग फील्ड होती है. क्लिनिकल ऑपरेशंस में आप कई प्रकार के जॉब कर सकते हो, जस्ट लाइक क्लिनिकल रिसर्च कोऑर्डिनेटर, क्लिनिकल रिसर्च एसोसिएट्स, प्रोजेक्ट मैनेजर्स. ये ऐसे रोल होते हैं और इसमें आपका बेसिकली काम होता है कोर रिसर्च पे काम करना. मतलब हमने एक नया मॉलिक्यूल जिसके बारे में हमें नहीं पता, ये बॉडी में जाने के बाद क्या इफेक्ट करेगा, ये हम पेशेंट को दे रहे हैं और अब देख रहे हैं कि क्या होने वाला है, और हम तैयार हैं कि जो कुछ भी पेशेंट के साथ होगा, हमें अगर वो गलत हुआ तो हमें उसे ठीक करना है कि नहीं. तो ये कितना डिफिकल्ट काम है. अब जो हो रहा है उसे डॉक्यूमेंट भी करना है एज एन एविडेंस, अदरवाइज मानेगा कौन कि ये ऐसा ऐसा हुआ था. तो क्लिनिकल रिसर्च ऐसी फील्ड है इस ऑपरेशन में जहां पे नए मॉलिक्यूल को पेशेंट को दिया जाता है अंडर द क्लिनिकल ट्रायल्स. इसमें काफी सारे प्रोसीजर होता है, जो अगर आप जैसे कोर्स ज्वाइन करोगे तो आपको पता चलेगा कि पेशेंट का कंसेंट लिया जाता है, उनके प्री मेडिकल्स चेकअप्स किए जाते हैं, उनके सारे सिस्टम्स को जाना जाता है, प्रीवियस डिजीज को जाना जाता है, एंड एज पर द प्रोटोकॉल एलिजिबिलिटी क्राइटेरिया को वेरीफाई किया जाता है. उसके बाद पेशेंट को वो मॉलिक्यूल दिया जाता है. तो मॉलिक्यूल देने के बाद भी कितने दिनों तक उनसे फॉलोअप लेना पड़ता है, सारी चीजें देखनी पड़ती है. उसके बाद हमें पता चलता है कि ये पेशेंट को ये ड्रग से कोई साइड इफेक्ट हुए या नहीं हुए, और इसको इफेक्ट आया या नहीं आया. सो डिस्कवरी ऑफ न्यू मॉलिक्यूल थ्रू क्लिनिकल ट्रायल इज नथिंग बट द क्लिनिकल रिसर्च. एंड you have so many career opportunities available in this field. जब आप ये कोर्स ज्वाइन करोगे तब आपको पता चलेगा. अब हम कुछ बात करते हैं कि ये क्लिनिकल ट्रायल जरूरी क्यों है. तो आप मुझे बताइए कि बिना क्लिनिकल ट्रायल कंडक्ट किए क्या कोई ऐसा तरीका है जिससे हम नया ड्रग्स इंट्रोड्यूस कर सकते हैं मार्केट में, बेटर ड्रग इंट्रोड्यूस कर सकते हैं मार्केट में? एनीबॉडी एल्स? क्या आपके पास कोई नया तरीका है? अगर नया तरीका होगा तो आप भी साइंटिस्ट बन सकते हैं. एनी न्यू वे विदाउट परफॉर्मिंग क्लिनिकल ट्रायल्स कैन वी इंट्रोड्यूस न्यू एंड बेटर मेडिसिन और ड्रग इनटू द मार्केट? ओके, प्रेजेंटेशन स्टार्ट हो गया है. एनीबॉडी एल्स? एनी आइडिया? सो सदियों से क्लिनिकल ट्रायल चल रहे हैं. सो आज की तारीख में क्लिनिकल ट्रायल्स आर द ओनली वे by which we can introduce or we can bring to the world new and better drug into the market. Recent example हमारे साथ था 5-6 साल पहले जब कोविड पेंडेमिक आया था. सारी फार्मास्यूटिकल्स कंपनियों के पास अपॉर्चुनिटीज आई थी कि हम इसके वैक्सीन्स बनाएं. इंडिया में 2 वैक्सीन काफी फेमस चल रहे थे. कौन से? कोवैक्सीन और कोविशील्ड. हमने, जो हमारी कंपनी है प्रेरणा क्लिनिकल रिसर्च, हमने नागपुर में एक तीसरी वैक्सीन पे ट्रायल किया था जो जाइडस कंपनी की थी, जाइकोविड. अब होता कैसा है देखो, अपॉर्चुनिटी तो सबके पास आई, ये कोविड का वैक्सीन बनाना है, एवरीबडी हैज अ अपॉर्चुनिटी टू मेक द वैक्सीन. अब बेटर वैक्सीन कौन बनाता है वही रेस में आगे बढ़ेगा. सही बात है. और बेटर वैक्सीन के साथ जो उसे इकोनॉमिक प्राइस में बेच पाएगा. लेकिन बेचना तो दूर की बात है, सबसे पहले बनाना जरूरी है. करेक्ट. तो चूंकि हमें ये पेंडेमिक को सॉल्व करना था, इसलिए जो वैक्सीन पहले बनता उसी को ही सबको दिला के हमको कोविड वैक्सीन से निकलना था. बट यही एग्जांपल आप दूसरी बीमारी में अप्लाई कर सकते हो. तो हुआ कैसा था कि जो कोविड वैक्सीन और कोवैक्सीन बने थे, ये लाइव वायरस थे जिनको इनएक्टिव किया गया था और बॉडी में डालने के बाद उसके एंटीबॉडीज बनाए गए. करेक्ट. ऐसा ही था. लेकिन हम जो जाइडस के वैक्सीन पे काम कर रहे थे उसमें लाइव वैक्सीन नहीं था. उन्होंने थोड़ा सा एडवांस सोचा. उन्होंने उसका जो आरएनए था, उस आरएनए को एक्सट्रैक्ट करके उसके अगेंस्ट में एंटीबॉडी बनाए थे. अब ये वैक्सीन इतना बढ़िया था. अब होता कैसा था कि जब हमारा ये कोविड वैक्सीन का फेज स्टार्ट हुआ तो गवर्नमेंट ने स्टार्ट किया कि सबसे पहले हम पुलिस वालों को, मेडिकल को और पैरामेडिकल स्टाफ को वैक्सीन देना चालू करेंगे. तो हमारे पास जब ये प्रोजेक्ट आया तो जिस हॉस्पिटल के स्टाफ को हम वैक्सीन देने वाले थे, ऑलरेडी उन्होंने वैक्सीन ले चुका था. तो अब ये वैक्सीन किसको देंगे? तो हमने कहा कि आप अपने फैमिली को सिक्योर कर सकते हैं, आप अपने फैमिली मेंबर को ये वैक्सीन दे सकते हैं. तो उनके फैमिली मेंबर को हमने वैक्सीन दिए. कोवैक्सीन, कोविशील्ड के 2-2 डोज होते थे, इसके 3 डोज हुआ करते थे 1-1 महीने की गैप में. 3 डोज लेने के बावजूद उनके जो मेडिकल स्टाफ थे हॉस्पिटल के, वो आके पूछने लगे आपने सच में वैक्सीन दिया भी है या नहीं दिया है? तो हमने ऐसे पूछा कि ऐसा क्यों बोल रहे आप? बोले 3 डोज हो जाने के बावजूद भी हमारे रिलेटिव्स को कुछ नहीं हो रहा है. तो मतलब ये अच्छी बात है ना? नहीं, हमें डाउट हो रहा है कि आपने वैक्सीन नहीं दिया है. क्यों? क्योंकि हमने जो वैक्सीन लिया है वो लेने के बाद हम तो 2 दिन घर में पड़े थे. किसी को हेडेक हो रहा है, किसी को बॉडी पेन हो रहा है, किसी को फीवर हो रहा है, लेकिन ये वैक्सीन से कुछ भी नहीं हो रहा है. जब इनके हमने ब्लड सैंपल के एंटीबॉडी टेस्टिंग किए तो बहुत ज्यादा एंटीबॉडीज निकले. तो इसका क्या मतलब हुआ है कि ये एंटीबॉडीज बने हैं, मतलब वैक्सीन ने काम किया है. लेकिन ये वैक्सीन इतना सेफ था कि पेशेंट को कुछ भी नहीं हुआ है. बराबर है. दैट इज एडवांस टेक्नोलॉजी. अब ये कैसे हुई? थ्रू क्लिनिकल ट्रायल्स. हालांकि कोविड उस समय फिर खत्म हो गया था, इसलिए ये वैक्सीन को बेचने की कोई जरूरत नहीं पड़ी. लेकिन ये बेटर वैक्सीन था दूसरे भी मायने में. कैसे? कोवैक्सीन और कोविशील्ड एडमिनिस्टर किए जाते थे ट्रेडिशनल टेक्निक सिरिंज से. करेक्ट है. ये वाला वैक्सीन दिया जाता था फार्माजेट डिवाइस से, इंट्राडर्मल, मतलब जो चमड़ी है अपनी उस पर ही वो वैक्सीन का दिया जाता था. एक डिवाइस होता था जिससे दे देते थे. आपको पता भी नहीं चलता था वैक्सीन है. जैसे वो फार्माजेट डिवाइस है, यहां लगाया, ऐसा छोड़ दिया, ये वैक्सीन हो गया है. तो बहुत से जन ऐसे बैठे रहते थे, उनको वैक्सीन हो जाता था, उनको पता भी नहीं चलता था कि उनको वैक्सीन लग गया है. हमको बताना पड़ता था आपका वैक्सीन हो गया, आप जा सकते हैं. तो उन्हें पता ही नहीं वैक्सीन लगा कि नहीं लगा. तो ये है एडवांस टेक्नोलॉजी. ये सारी की सारी एडवांस टेक्नोलॉजी बन तो जाती है, लेकिन मार्केट में आने के लिए सबसे पहले उस पर ट्रायल करना जरूरी है. अदरवाइज हम कैसे प्रूव करेंगे कि ये इफेक्टिव है और सेफ है? उसी के लिए हमें क्लिनिकल ट्रायल्स करने पड़ते हैं. नाउ विल गो बैक टू द पीपीटी. कुछ छोटे से स्लाइड्स हैं जो मैं आपके सामने रखना चाहता हूं. जैसे कि यहां पे कहा है कि व्हाट इज क्लिनिकल रिसर्च. क्लिनिकल रिसर्च नथिंग बट द ड्रग डिस्कवरी. कैसे की जाती है, सभी लोग अपने अपने अलग मेथड यूज करेंगे, सभी लोग अपने अपने अलग सोच लाएंगे, अपनी अपनी टेक्नोलॉजीज को इन्वॉल्व करेंगे, हर एक कंपनी टेक्नोलॉजीज लाएंगी। तो कैसे भी करके हमें सिर्फ दो चीज को प्रूव करना है। कौन सी दो चीज? कौन सी दो चीज प्रूव करना है? करेक्ट। कैसे भी करके हमें हमारे मॉलिक्यूल की सिर्फ दो चीजें प्रूव करना है, वो है सेफ्टी और दूसरा एफिकेसी। अगर ये आपने प्रूव कर लिया तो आपका ड्रग मार्केट में सेल होने के लिए रेडी है। जस्ट लाइक अ man-made molecule becomes drug or medicine for a particular disease, either for treatment or prophylaxis, or a scientific study of how a new medicine or treatment works in a people. नीड ऑफ क्लिनिकल रिसर्च, अभी मैंने ऑलरेडी बता दिया। नए मेडिसिन की कितनी जरूरत है? फॉर एग्जांपल कोविड वैक्सीन, कोविड आया तो हमें नए वैक्सीन की जरूरत पड़ी। ऐसे ही कई बीमारियां आ रही हैं और बहुत सी एग्जिस्टिंग डिजीज है जिसके लिए अभी भी बेटर और इफेक्टिव मेडिसिन मार्केट में अवेलेबल नहीं है। सो दिस इज द स्कोप फॉर यू पीपल टू गो एंड डेवलप अ न्यू एंड बेटर ड्रग। अंडरस्टैंड? मैं कुछ लिखे, क्लिनिकल रिसर्च इज बाय टू सेफ एंड इफेक्टिव मॉलिक्यूल इन द मार्केट। सेफ्टी एफिकेसी प्रूव करने के लिए क्लिनिकल ट्रायल करना पड़ता है रेगुलेटरी अप्रूवल के लिए। बिना रेगुलेटरी अप्रूवल आप मार्केट में ड्रग बेच नहीं सकते, दैट इज इलीगल। हालांकि बहुत सारे लिखते हैं रेगुलेटरी अपने हिसाब से उनको कंट्रोल कर रही है, बट लीगली बिना उनके अप्रूवल के आप ड्रग नहीं बेच सकते। और ये अप्रूवल क्यों जरूरी है? ये अप्रूवल इसलिए जरूरी है, इफ वी गेट द अप्रूवल फ्रॉम द रेगुलेटरी वी कैन कॉन्फिडेंटली सेल अवर मॉलिक्यूल टू द पीपल। क्योंकि रेगुलेटरी तभी अप्रूवल देगी जब आप उनके सामने एविडेंस पुट करोगे कि हमने इस पर्टिकुलर मॉलिक्यूल पे सीरीज ऑफ क्लिनिकल ट्रायल्स किए हैं और सारे के सारे क्लिनिकल ट्रायल्स में पॉजिटिव रिजल्ट आए हैं। वी हैव गॉट अ गुड सेफ्टी प्रोफाइल एज वेल एज एफिकेसी प्रोफाइल। तभी रेगुलेटरी आपको आपके मॉलिक्यूल के लिए अप्रूवल देगी, अदरवाइज वो नहीं देगी। तो रेगुलेटरी अप्रूवल हमें कॉन्फिडेंस देता है एज वेल एज लीगल लाइसेंस देता है कि आप अपना मॉलिक्यूल मार्केट में बेच सकते हैं। टू फाइंड आउट सेफ एंड इफेक्टिव ड्रग, प्रोटेक्ट ट्रायल सब्जेक्ट, ये बहुत इंपॉर्टेंट चीज है। कई सालों पहले जब युद्ध हुआ करते थे, अभी तीसरा वर्ल्ड वॉर चालू है लेकिन 2-3 कंट्री लड़ रही है। पहले जमाने में, हिटलर के जमाने में जब एकदम खतरनाक वॉर हुआ करते थे, उस समय भी क्लिनिकल ट्रायल्स हुआ करते थे, लेकिन सारे अनएथिकल। गुलाम बना दिया है, अब ये गुलाम लोग हैं, इन पे जो मर्जी आए वो क्लिनिकल ट्रायल करो। कुछ कुछ ट्रायल मैं आपको बताता हूं कैसे हुआ करते थे। जैसे कि पहाड़ी में, ऊंचाई में हमको युद्ध लड़ना है, ऊपर ऑक्सीजन कम रहती है, तो ऐसे कम ऑक्सीजन में भी हमारे सोल्जर्स जिंदा रहे और इफेक्टिवली लड़े, उसके लिए ट्रायल किए जाते थे। तो वो ट्रायल किस पे? सोल्जर्स पे नहीं किए, वो गुलामों पे भी है, बिना उनका कंसेंट लिए कि हम आपको कुछ ऐसी चीज दे रहे हैं जिसके बारे में हमको खुद नहीं मालूम कि ये काम करेगी कि नहीं करेगी, लेकिन आप पे डालेंगे, आप मर गए तो कोई फर्क नहीं पड़ता। फिर जैसे समंदर में ट्रायल करते हैं, अभी समंदर में वॉर चालू है और उनको पानी की जरूरत पड़ी, तो समंदर का पानी खारा होता है, नमक वाला, वो पी सकते कि नहीं पी सकते, उस पर ट्रायल किए जा रहे हैं। वो सी वाटर के इंजेक्शन गुलामों को दिए जाते थे, प्रिजनर्स को दिए जाते थे, वो पूरा डिहाइड्रेट हो जाती थी, पूरी बॉडी मर जाते थे। लाखों लोग मारे गए अनएथिकल तरीके से। तो ऐसे अनएथिकल ट्रायल्स नहीं होने चाहिए, इसलिए रेगुलेटरी गवर्नमेंट ने क्लिनिकल ट्रायल्स कंडक्ट करने के लिए गाइडलाइंस बनाई है, रेगुलेशंस बनाए हैं, रूल्स बनाए हैं। यही सारे रूल्स, रेगुलेशन, गाइडलाइंस आप जब कोर्स ज्वाइन करेंगे तब आपको इसमें पढ़ने को मिलेंगे, और इनके थ्रू ही अगर ट्रायल किया जाएगा तो ही उसे एथिकल क्लिनिकल ट्रायल किया जाएगा। इसके इन रूल्स रेगुलेशन को फॉलो किए बगैर अगर कोई ट्रायल किया जाता है तो उस ट्रायल के सारे एविडेंसेस को, सारे रिजल्ट को डिनाई किया जाता है, निगलेक्ट किया जाता है। और एक बार रेगुलेटरी ने आपके रिजल्ट्स को डिनाई कर दिया, निगलेक्ट कर दिया, तो अब आप अपना मॉलिक्यूल मार्केट में नहीं बेच सकते। इसलिए ये सारे गाइडलाइंस, रेगुलेशंस फॉलो करने रहते हैं, और वो कैसे फॉलो करना है ये आपको कोर्स में बताया जाता है। टू जनरेट एथिकल एंड ट्रू डाटा, दैट इज जो अभी मैंने कहा। एथिकल डाटा कब रहेगा? जब आप गाइडलाइंस को फॉलो करोगे। टू डिफरेंशिएट बेटर ड्रग फ्रॉम द बैड ड्रग। इंडिया में क्लिनिकल ट्रायल कंडक्ट करना क्यों चाहिए? सबसे बड़ी बात यह है कि हमारे पास जनसंख्या का बहुत बड़ा क्या बोलते खजाना है और बीमारियों का भी बहुत बड़ा खजाना है। सारे प्रकार की दुनिया की जितनी बीमारियां हैं, सारी बीमारियां हमारे इंडिया में है और हमारे पास इतना पेशेंट पॉपुलेशन है कि एक दिन में जाओ तो 4-4 जन मिल जाते हैं, बराबर है। ऊपर से पुअर पॉपुलेशन होने के कारण पेशेंट को जरूरत रहती है फ्री ट्रीटमेंट की। क्लिनिकल ट्रायल्स में पूरा ट्रीटमेंट फ्री होता है। आज इंडिया में जितने भी क्लिनिकल ट्रायल्स हो रहे हैं, मोर देन 50% क्लिनिकल ट्रायल्स आर ऑन ऑन्कोलॉजी मेडिसिन, कैंसर मेडिसिन पे। और आप सबको पता है कि कैंसर की दवा कितनी महंगी रहती है, एक एक इंजेक्शन लाखों रुपए का होता है। ये सारे इंजेक्शन पेशेंट को फ्री में मिलते हैं। आजकल कैंसर गरीबों अमीरों की नहीं है, ये गरीबों में भी आती है। आज अगर आप गवर्नमेंट मेडिकल कॉलेज में जाओगे या कोई भी कैंसर हॉस्पिटल में आप जाओगे, आप देखोगे भीड़ पड़ी है कैंसर की। मतलब इतने सारे कैंसर पेशेंट है कि पूछो नहीं। तो इतने सारे कैंसर पेशेंट में इतने गरीब लोग रहते हैं, तो वो इतनी महंगी दवाई अफोर्ड नहीं कर पाते। और कैंसर आज की तारीख में भी इतना इजी नहीं है कि सर्दी खांसी जैसा कि खा लिया तो पूरा कैंसर चले जाएगा। There is a huge scope available in the field of development of cancer medicines. तो कैंसर मेडिसिन डेवलप करने के लिए काफी स्कोप है, इसलिए 50% से ज्यादा क्लिनिकल ट्रायल कैंसर पे ही होते हैं। तो इंडिया में क्लिनिकल ट्रायल करना क्यों चाहिए या क्यों आसान है? तो यही एक कारण है कि हमारे पास पेशेंट पॉपुलेशन ज्यादा है, हमारे पास डिजीज भी पेशेंट पॉपुलेशन बहुत ज्यादा है। अपार्ट फ्रॉम दैट, इंडिया में अभी आप देखें ना, नागपुर सिटी में आप सब लोग रहते हो, अगर आप नागपुर में घूमेंगे ना, आज की तारीख में 10 से 15 बड़े बड़े हॉस्पिटल बन रहे हैं, समझ ना? कोई 50 बेड का, कोई 100 बेड का, कोई 200 बेड का, कुछ गवर्नमेंट वाले अपना एक्सटेंशन कर रहे हैं। इतने सारे हॉस्पिटल बन रहे हैं और सारे के सारे हॉस्पिटल चल रहे हैं। और इतने बड़े बड़े हॉस्पिटल्स में काफी अच्छा इंफ्रास्ट्रक्चर अवेलेबल है हर कोई बीमारी को ठीक करने के लिए। तो जो फॉरेन फार्मास्यूटिकल्स कंपनियां रहती है, उनको चाहिए रहता है कि अच्छा इंफ्रास्ट्रक्चर होना चाहिए क्लिनिकल ट्रायल कंडक्ट करने के लिए। जैसे कि अगर आप शादी करने जाओगे तो देखोगे ना, अच्छी लड़की होनी चाहिए, अच्छा लड़का होना चाहिए, खानदान अच्छा होना चाहिए, हाँ, दुकानें होनी चाहिए या बिजनेस होना चाहिए। वैसे वो फॉरेन फार्मास्यूटिकल कंपनियां भी यही देखती है कि यहाँ पे पेशेंट पॉपुलेशन तो है, लेकिन क्या वो इंफ्रास्ट्रक्चर अवेलेबल है, क्या वो मशीन्स अवेलेबल है जिसके थ्रू हमको क्लिनिकल ट्रायल करना है कि नहीं। तो आज की तारीख में इंडिया के पास वो सारी चीजें भी अवेलेबल है और काफी हॉस्पिटल्स क्लिनिकल रिसर्च और क्लिनिकल ट्रायल पर फोकस करते हैं, जिसमें जो भी मिसिंग इंफ्रास्ट्रक्चर रहता है वो मिसिंग इंफ्रास्ट्रक्चर को भी पूरा करते हैं ताकि ज्यादा से ज्यादा क्लिनिकल ट्रायल्स कर सके। These are the other I think reasons कि इंडिया में, वैसे तो बहुत सारे रीजंस हैं, लेकिन मैंने अभी आपको कुछ रीजंस बताए जिससे इंडिया में क्लिनिकल ट्रायल करना। अच्छा, और एक रीजन, जो क्लिनिकल ट्रायल यूएस में, यूरोप में 100 रुपए में होगा, वो इंडिया में 20-25 रुपए में हो जाएगा। यह और एक बहुत बड़ा रीजन है कि इंडिया में कोई भी क्लिनिकल ट्रायल करना हो तो बहुत सस्ते में होता है, क्योंकि वहां के जो रूल्स रेगुलेशन है जिनके कारण उनको काफी ज्यादा पैसे लगते हैं, इंडिया में नहीं लगते। अब ये बहुत इंपॉर्टेंट स्लाइड है आप लोगों के लिए, हु इज इंटरेस्टेड टू नो अबाउट व्हाट इज क्लिनिकल रिसर्च। दिस इज व्हाट इज द ड्रग डेवलपमेंट प्रोसेस। कैसे एक मॉलिक्यूल जो रिसर्च लैब में बनाया है, वो मॉलिक्यूल ड्रग और मेडिसिन में कन्वर्ट होता है। तो सबसे पहले क्या होता है, R&D। हर एक फार्मास्यूटिकल कंपनी के पास अपना एक R&D सेटअप होता है। उस R&D सेटअप में कई डॉक्टर्स, कई साइंटिस्ट कंटिन्यूअसली लगे रहते हैं, कंटिन्यूअसली अपना पूरा नॉलेज लगाते हैं, अपना पूरा एक्सपीरियंस लगाते हैं कि हम ऑलरेडी जो ड्रग अवेलेबल है उससे बेटर ड्रग ढूंढ के निकाले। ऑलरेडी जो ड्रग अवेलेबल है जिसमें जो साइड इफेक्ट हो रहे हैं, उन साइड इफेक्ट को और मिनिमाइज करें। तो ऐसे हजारों मॉलिक्यूल वो बनाते हैं, लेकिन उन मॉलिक्यूल में से कोई 2 या 3 मॉलिक्यूल ही प्रॉमिसिंग रिजल्ट देते हैं। वो मॉलिक्यूल्स नेक्स्ट स्टेज जाते हैं प्री क्लिनिकल ट्रायल्स. प्री क्लिनिकल ट्रायल्स नथिंग बट द ट्रायल्स ऑन एनिमल. अंडरस्टैंड? क्लिनिकल ट्रायल्स मीन ट्रायल्स ऑन ह्यूमन बीइंग, प्री क्लिनिकल ट्रायल्स मतलब ट्रायल्स ऑन एनिमल. प्री का ही फर्क है. प्री मतलब एनिमल पे, क्लिनिकल मतलब ह्यूमन पे. अब ह्यूमन क्लिनिकल ट्रायल्स जो होते हैं वो 4 फेज में होते हैं. ठीक है? फेज 1, फेज 2, फेज 3, फेज 4. हर एक फेज पे अलग अलग ऑब्जेक्टिव्स रहते हैं. जब हम फेज 1 ट्रायल करते हैं तब पहला एक ही ऑब्जेक्ट रहता है, सेफ्टी. सबसे पहले हमारा जो मॉलिक्यूल है वो सेफ होना चाहिए. तो पूरे का पूरा प्रोटोकॉल, पूरे के पूरे सारी चीजें सेफ्टी को डिटरमाइन करने में लगती है कि अभी हम सबसे पहले ये जानवरों के बाद हम सबसे पहले इंसानों को ये ड्रग दे रहे हैं. तो अब इंसानी बॉडी में क्या हो रहा है, फार्माकोडायनामिक्स, फार्माकोकाइनेटिक्स, सारी चीजें चेक की जाती है. मैक्सिमम टॉलरेटेड डोज, मिनिमम टॉलरेटेड डोज, मिनिमम इफेक्टिव डोज, ये सारी चीजें चेक की जाती है. ये और बहुत कम पॉपुलेशन में, ताकि बहुत ज्यादा पॉपुलेशन को रिस्क में हम न डालें. सेफ्टी डेटा आने के बाद हम जाते हैं फेज 2 में. अब फेज 2 में पहली बार ये मेडिसिन पेशेंट को दी जाती है जिस बीमारी के लिए बनाई गई है. समझ लो कार्डियक डायबिटीज के लिए बनाई गई है, तो सबसे पहली बार डायबिटीज पेशेंट कहां आएगा? फेज 2 में आएगा. फेज 1 में हमेशा हेल्दी वॉलंटियर्स रहते हैं, कुछ कुछ मेडिसिन को छोड़ सके. तो फेज 2 में हम फिर से सेफ्टी के साथ साथ अब पहली बार एफिकेसी पे भी ध्यान देते हैं. फेज 2 में सेफ्टी और एफिकेसी के पॉजिटिव रिजल्ट्स आने के बाद अब लार्ज पॉपुलेशन में हम फेज 3 में ट्रायल करते हैं. फॉर एग्जांपल 50 सब्जेक्ट्स अगर हमने फेज 1 में लिए थे तो फेज 3 पहुंचते पहुंचते वो 500 हो जाते हैं. अंडरस्टैंड? फेज 3 में अब प्राइमरी ऑब्जेक्टिव होता है एफिकेसी, क्योंकि फेज 1 और फेज 2 में हमने सेफ्टी प्रोफाइल काफी डेटा कलेक्ट कर लिया है. बट इट डज नॉट मीन कि अभी भी हमको सेफ्टी पे ध्यान नहीं देना है. फेज 3 में भी सेफ्टी पे ध्यान दिया जाता है, लेकिन अब फेज 3 में एफिकेसी इज द प्राइमरी ऑब्जेक्टिव. फेज 3 में एफिकेसी और सेफ्टी के पॉजिटिव रिजल्ट आने के बाद अब ये ड्रग मार्केट में लॉन्च होने के लिए रेडी है. अब रेगुलर जो भी फार्मास्यूटिकल्स कंपनियां होती है अब इसको मार्केटिंग करती है. बट कहानी अभी भी यहां पे खत्म नहीं होती है. फेज 3 के बाद फेज 4 क्लिनिकल ट्रायल्स किए जाते हैं, जिसे पोस्ट मार्केटिंग सर्विलेंस भी कहा जाता है. अभी सर ने हमारे इस मेरे लेक्चर से पहले काफी डिटेल में आपको इसके बारे में समझाया है. इसमें फेज 4 भी ट्रायल किए जाते हैं, फार्माकोविजिलेंस भी इंक्लूड होता है. चूंकि ऑलरेडी ड्रग मार्केट में आ गया है, लेकिन ये तो सिर्फ 400-500 लोगों पे किया गया है. अभी तो 400-500 करोड़ मतलब 400-500 लाख लोग इसको खाने वाले हैं. तो उनके साथ क्या होने वाला है, तो उनका सेफ्टी डेटा भी कलेक्ट करना जरूरी है. गवर्नमेंट काफी इनिशिएटिव्स लेती भी है, इवन फेज 4 ट्रायल्स भी होते हैं कि हम ये सेफ्टी का डेटा ज्यादा से ज्यादा कलेक्ट कर सके. बट अभी भी उतना स्ट्रांग इंफ्रास्ट्रक्चर नहीं है इंडिया में जितना फॉरेन में हुआ करता है. तो अभी भी उस पर डेवलपमेंट चालू है, शायद हम भी उस पर अच्छा ही काम करेंगे, गवर्नमेंट करेगी. बट ये फेज 4 ट्रायल चलते हैं, 5, 6, 7 साल के बाद हमें लगभग सारे एडवर्स इवेंट जो भी मोस्ट ऑफ होने वाले रहते हैं सब पता चल जाते हैं. अगर कोई ड्रग 5 साल 6 साल तक मार्केट में चल जाता है और अच्छा रिजल्ट देता है, तो समझ लो ये ड्रग अभी आगे मार्केट में चलने वाला है. तो अभी आपने देखा कहां एक लैब में बना हुआ मॉलिक्यूल अभी मार्केट में एज ए ड्रग सेल आउट हो रहा है. बराबर है? अब इसके अलावा कुछ ट्रायल्स होते हैं जैसे फार्माको पीकेपीडी स्टडीज और बायोमेडिकल हेल्थ रिसर्च. पीकेपीडी स्टडीज नथिंग बट द जेनेरिक ड्रग बनाने के लिए होते हैं, और हमारे इंडिया में ये बहुत ज्यादा होते हैं क्योंकि हमको कॉपी करने की बहुत आदत है. बराबर है? नए ड्रग्स बनाते हैं फॉरेन वाले, हमारे वाले क्या सोचते हैं पता है? क्यों नया ड्रग बनाने का, इतना दिमाग क्यों लगाने का? वो क्या करते हैं, जो भी नया ड्रग यूरोप में या अमेरिका में लॉन्च हुआ है बस उस पर फोकस करो. 5 साल के बाद उसका पेटेंट खत्म हो जाएगा. अगर ये ड्रग वहां पे बहुत बढ़िया चल रहा है तो 5 साल के बाद हमें उसकी कॉपी करना है और सीधा फार्मा ये पीकेपीडी स्टडीज करना है और मार्केट में लॉन्च कर देना है. रिसेंट एग्जांपल्स हम बताते हैं. एक एग्जांपल मैं बताता हूं, एडवांस रीनल सेल कार्सिनोमा. यह होता है किडनी का कैंसर, और एडवांस मतलब किडनी निकालने के बाद भी वो दूसरी जगह फैलता है, लंग्स में फैलता है, आंतड़ियों में फैलता है, ब्लैडर में जाता है। तो इसका एक ड्रग था, वो ड्रग जब मार्केट में लॉन्च हुआ था तब 10 टैबलेट का एक पत्ता हुआ करता था। वो 10 टैबलेट का एक पत्ता 1,80,000 का था। कितने का? 1,80,000 का। अब उसका पेटेंट एक्सपायर हो गया। अब इंडियन कंपनी ने उसको बनाना चालू कर दिया। चालू करने के बाद उसके रिजल्ट आ गए। उनको सिर्फ ये पीके प्रोफाइल दिखाना रहता है कि ये इनोवेटर का मॉलिक्यूल खाया और ये हमारा मॉलिक्यूल खाया। अब दोनों में इस पर्टिकुलर टाइम के बाद जो अवेलेबिलिटी ऑफ मॉलिक्यूल है, इसका भी ग्राफ ये है, इसका भी ग्राफ ये है, नो सिग्निफिकेंट डिफरेंस, सो यू कैन नाउ मैन्युफैक्चर इट। तो ये मेडिसिन उस समय कितने की हुई? एक गोली 18,000 की। खाते खाते गलती से बेसिन में गिर गए तो 18,000 गए, बराबर ना? खाते खाते किसी ने धक्का दे दिया, बच्चे ने, गोली बेसिन में गई, 18,000 घुस गए। अब निकाल भी नहीं सकते आप। यही मेडिसिन उन्होंने बाद में 2,000 रुपए का पत्ता निकाले। कितने का? 2,000 का। और एक रिसेंट एग्जांपल देता हूं, आप नेट पे सर्च कर सकते हैं, सिजोफ्रेनिया करके एक डिजीज होती है, पागलपन से थोड़ी सी नीचे, बराबर है? उसके लिए मंथली इंजेक्शन आता है जो जॉनसन एंड जॉनसन कंपनी ने बनाया है, उसका नाम है इनवेगा सस्टेना। वो मॉलिक्यूल 1 मंथ का 10,700 रुपए का है इंजेक्शन। कितने का? 10,700 का। हमने 2022 में जीएमसी में क्लिनिकल ट्रायल किया सन फार्मा का, सेम मॉलिक्यूल पे, और वो मॉलिक्यूल आज अवेलेबल है मार्केट में। अब सन फार्मा उसको बेचता है 1,500 रुपए में। कितने में? 1,500 रुपए में। जॉनसन एंड जॉनसन ने सन फार्मा पर केस किया, वो जो चल रहा है चलने दो, तब तक ये बहुत सारा बेच के अपना कमा लेगा। लेकिन 10,000 का इंजेक्शन कितने में आया? 1,500 में। और इस पे और क्लिनिकल ट्रायल्स अभी भी रिसेंटली हम कर ही रहे हैं नागपुर में। आने वाले दिनों में 500 रुपए का हो जाएगा। बराबर है? तो होता क्या है कि इनोवेटर इतना महंगा क्यों बेचता है? कोई बता सकता है? इतना महंगा क्यों बेचता है? हा? क्या बोले? एक्सेस। क्या होता है, क्लिनिकल ट्रायल्स करना, यानी ये जो नया मॉलिक्यूल बनाना है, इसमें बहुत पैसा लगता है। इसमें साइंटिस्ट लोगों का भी तो पैसा लगता है, प्लस जो क्लिनिकल ट्रायल्स में जो डॉक्टर्स इन्वॉल्व रहते हैं, इन्वेस्टिगेटर, उन लोगों को भी काफी पेमेंट जाता है, काफी पैसा मिलता है। ये पैसा निकालेंगे कहां से? तो इनको जो पेटेंट दिया जाता है 5 साल, 7 साल, 10 साल का, ये केस में क्या करते हैं, वो उनका रेट इतना हाई रखते हैं कि सारा का सारा पैसा निकल जाता है। और आप गौर करना, एक फार्मास्यूटिकल कंपनी के पास सैकड़ों हजारों मॉलिक्यूल्स होंगे, लेकिन वो 2-4 मॉलिक्यूल उसके मार्केट में चल रहे होंगे, और वो 2-4 मॉलिक्यूल से पूरी कंपनी चल रही है, बाकी तो ड्रग उसके बिक ही नहीं रहे। समझिए, ये 2-4 मॉलिक्यूल से पूरी कंपनी चल रही है। तो देखने वाली बात है कि इतने महंगे, आज एक गोली, छोटी सी गोली 100 रुपए की मिलती है, क्या उसको बनाने में 100 रुपए लगते हैं? ये पूरा रिसर्च का पैसा है। किसका पैसा है? रिसर्च। तो आप सोचिए कि जब आप इस फील्ड में आएंगे क्लिनिकल रिसर्च में, तो आपके लिए कितना हैंडसम सैलरी पैकेज होगा, क्योंकि ये आपका सैलरी भी ये मेडिसिन पे लेता है और ये जनता पे करती है। लेकिन जब जेनेरिक ड्रग बनाए जाते हैं, इसलिए हमारे गवर्नमेंट जेनेरिक ड्रग को क्यों प्रमोट कर रहे हैं? क्योंकि सारा इनोवेशन का पैसा कौन लगाएगा? फॉरेन की कंपनियां। हमें बस इंतजार करना है पेटेंट एक्सपायर होने का। पेटेंट एक्सपायर हुआ कि हम चालू करेंगे जेनेरिक ड्रग बनाना और उसको सस्ते सस्ते में बेचेंगे। ठीक है? अच्छा, बायोमेडिकल हेल्थ रिसर्च, ये कैसा रहता है? आईसीएमआर का नाम सुना है आपने? इंडियन काउंसिल ऑफ मेडिकल एजुकेशन। आईसीएमआर क्या करती है कि कुछ रिसर्च प्रोग्राम इनिशिएट करती है जिसमें ड्रग नहीं रहता है, बस ऑब्जर्वेशनल स्टडीज रहती है कि फॉर एग्जांपल ये पर्टिकुलर डिजीज है, इस पे ये मेडिसिन अगर ले रहे हैं तो क्या इफेक्ट आ रहा है। तो उनकी कुछ चेकलिस्ट रहती है, वो चेकलिस्ट के हिसाब से डेटा कलेक्ट किया जाता है और डेटा कलेक्ट करने के बाद उनका एनालिसिस होता है और एनालिसिस करने के बाद वो कुछ अपना कंक्लूजन निकालते हैं। तो ऐसी स्टडीज को बोलते हैं बायोमेडिकल हेल्थ रिसर्च। इसमें कोई भी प्रकार का रिस्क इन्वॉल्व नहीं रहता है। इसी को जनरली हम क्लिनिकल ट्रायल में इंक्लूड नहीं करते। क्लिनिकल ट्रायल हम उसी बोलेंगे जिसमें नया मॉलिक्यूल इन्वॉल्व हो, जिसपे हम एक्चुअली डेटा जनरेट कर रहे हो। दीज आर द फील्ड्स जो मैंने इनिशियली आपको बताई। क्लिनिकल रिसर्च में 6 प्रकार के डिफरेंट फील्ड्स हैं, और इस करियर की ब्यूटी क्या है मैं बताता हूं। अगर आप क्लिनिकल रिसर्च ऑपरेशन में काम कर रहे हो ना, 2-3 सालों के बाद आपकी इच्छा हुई कि मुझे डेटा मैनेजमेंट में जाना है, तो उसी नॉलेज के साथ आप डेटा मैनेजमेंट में जा सकते हो। आपकी इच्छा हुई कि मुझे रेगुलेटरी में जाना है, तो उसी एक्सपीरियंस के साथ आप रेगुलेटरी में भी जा सकते हो। अगर आपको लगे कि मुझे फार्माकोविजिलेंस में जाना है, तो उसी एक्सपीरियंस के साथ आप फार्माकोविजिलेंस में भी जा सकते हो। यानी इनिशियल लेवल पे अगर आपको कहीं पे भी स्विच करना हो तो आप बहुत आसानी से स्विच कर सकते हैं। दिस इज द ब्यूटी ऑफ क्लिनिकल रिसर्च। अभी आप लोग सोचोगे कि क्लिनिकल रिसर्च करना है तो हमें क्या करना है, तो आप लोगों को इस 2 प्रकार की चीजें अपने अंदर में लाना है। एक साइड है टेक्निकल और दूसरी साइड है मैनेजरियल। आपके पास टेक्निकल नॉलेज भी होनी चाहिए और आपके पास मैनेजरियल नॉलेज भी होनी चाहिए। टेक्निकल नॉलेज इनिशियल फेज ऑफ जॉब में बहुत ज़्यादा जरूरी होती है, लेकिन जैसे जैसे आप एक्सपीरियंस लेते जाते हो और आगे बढ़ते जाते हो, आपके अंदर में मैनेजरियल स्किल्स बढ़ना जरूरी हो जाता है। तो टेक्निकल स्किल्स क्या है? गाइडलाइंस, जो मैंने रेगुलेशंस और गाइडलाइंस बताए, उन सब के बारे में नॉलेज होना चाहिए। फिर एनाटॉमी फिजियोलॉजी ऑफ ह्यूमन बॉडी, वो तो आपने ऑलरेडी 12th में पढ़ा ही होगा, फिर से रिवाइज कर लेना। देन होता है डिजीजेस, बीमारियों के बारे में आपको पता होना चाहिए, जो कि आपको बहुत आसानी से पता हो सकता है, क्योंकि आजकल मतलब आप जब मेडिसिन पढ़ भी रहे हो तब भी आपको बीमारियों के बारे में पता हो रहता ही है। ड्रग रिलेटेड डिटेल, इसके बारे में तो ऑलरेडी आपको बहुत ज्यादा पता है। ये हो गई टेक्निकल स्किल्स। अब मैनेजरियल स्किल्स कौन सी है? आपको आपके जो भी टास्क रहते हैं उसको मैनेज करते आना चाहिए। सेकंड, बॉडी लैंग्वेज। जब आप मैनेजर बनते हो तब आपको आपकी टीम को लीड करना होता है। जब आपको आपकी टीम को लीड करना होता है तब आपका बॉडी लैंग्वेज, आपका कम्युनिकेशन, ये बहुत ज्यादा इंपॉर्टेंट है, क्योंकि अब आपको काम नहीं करना है, काम करेगी आपकी टीम। तो आपके तरफ से आपको उसको मोटिवेशन देना पड़ता है, अपनी एनर्जी उनके अंदर में डालनी पड़ती है, ताकि खच्चर भी घोड़ा बन जाए। समझे ना? जो ठंडा बैठा है उसके अंदर में भी जोश आ जाए और जम के काम करे। तो ये मैनेजरियल स्किल, लीडरशिप स्किल आपके अंदर में होनी चाहिए। प्रोफेशनलिज्म हमारे इंडिया के लिए बहुत ज्यादा जरूरी है, नॉट ओनली इन द क्लिनिकल रिसर्च फील्ड, हर एक फील्ड में हम प्रोफेशनलिज्म जितना ज्यादा अपने अंदर में लाएंगे, हमारे काम की एफिशिएंसी उतनी ही ज्यादा बढ़ेगी। प्रोफेशनलिज्म का मतलब यह होता है कि आपकी जो जवाबदारी है, आपकी जो रिस्पॉन्सिबिलिटी है, उसको ईमानदारी से करना, और उसमें अगर कुछ प्रॉब्लम आती है तो उसे सॉल्व करना, ना कि बैठ जाना, ये मेरे को नहीं जम रहा है, ये मेरा काम नहीं है। ये सारी बातों को दूर करके अपने अंदर में प्रोफेशनलिज्म लाना। और सबसे मेन चीज, इनिशिएटिव लिया कीजिए। अगर आपने ये काम खत्म कर लिया तो अब मुझे नया क्या करना है, नया क्या सीखना है, इसके लिए इनिशिएटिव लेना बहुत ज्यादा जरूरी है। देन पॉजिटिव एटीट्यूड, दिस इज नथिंग बट द पॉजिटिव एटीट्यूड। आप एक जगह पे स्टिक नहीं रह सकते। आपने कोई एक बेंचमार्क अगर अचीव कर लिया है, एक नॉलेज अचीव कर लिया है, एक एक्सपीरियंस अचीव कर लिया है, तो आगे के लिए तैयारी कीजिए। सही बात है? अगर कोई 100 मीटर की रेस दौड़ लेता है तो उसे और आगे की रेस के लिए तैयारी करनी चाहिए। सही बात है? तभी तो आप आगे बढ़ पाओगे। तो जैसे जैसे आप अपने करियर में आगे बढ़ते हो, आपको इस तरह की मैनेजरियल स्किल्स अपने अंदर में डेवलप करनी पड़ती है, और ये कहीं पे भी कोर्स वोर्स में सिखाई नहीं जाती। बाजू वाली सिखाई जाती है, इधर वाली नहीं सिखाई जाती। ये इनबिल्ट रहती है हर एक आदमी में, इसलिए मैनेजर हमेशा एक ही रहता है और एम्प्लॉय ज्यादा रहते हैं। समझ गए? तो आप में से जो इस स्किल्स को जल्दी से डेवलप करेगा वो लीडर या मैनेजर बनेगा। ये कुछ इंपॉर्टेंट प्लेयर्स रहते हैं, जैसे जो फार्मास्यूटिकल्स कंपनी रहती है, हम उनको क्लिनिकल टर्म की टर्मिनोलॉजी में स्पॉन्सर कहते हैं। स्पॉन्सर मतलब कौन होता है? पैसे देने वाला, बराबर ना? जो पैसे लगाएगा उसे हम क्या बोलेंगे? स्पॉन्सर। तो फार्मास्यूटिकल कंपनी जिसका मॉलिक्यूल है वो पूरा का पूरा खर्चा बियर करता है, इसलिए उसको बोला जाता है स्पॉन्सर। सीआरओ, अब क्या होता है बहुत बार, फॉर एग्जांपल आपको अपना घर बनाना है, लेकिन आपको घर बनाते नहीं आता, तो आप क्या करोगे? आर्किटेक्ट हायर करोगे। आर्किटेक्ट इज ए स्पेशलाइज्ड पर्सन, प्रोफेशनल, उसको पता है घर बनाना है। घर किसका है? आपका, लेकिन बना कौन रहा है? आर्किटेक्ट। तो सीआरओ नथिंग बट द वो है। इंडिया में ट्रायल करना है, लेकिन स्पॉन्सर का कोई भी यहाँ पे ऑफिस नहीं है, उनके कोई एम्प्लॉय नहीं है, कोई नहीं है, बट उनको इंडिया में क्लिनिकल ट्रायल करना है तो सीआरओ को हायर करते हैं। तो मॉलिक्यूल है स्पॉन्सर का, लेकिन ट्रायल कंडक्ट करेगा सीआरओ। सारे रेगुलेटरी परमिशंस, क्लिनिकल ट्रायल, ये सब कुछ वो करेगा और पूरा सब कुछ रेडी करके, जैसे आर्किटेक्ट चाबी देता है ना, आपका घर रेडी है, अब आप यहाँ पे रहने जा सकते हैं, वैसे सीआरओ पूरा क्लिनिकल ट्रायल कंडक्ट करके उनको पूरी रिपोर्टिंग करता है कि आपका क्लिनिकल ट्रायल कंप्लीट हो चुका है, ये उसका रिजल्ट है। रेगुलेटरी बॉडी इज नथिंग बट द गवर्नमेंट, जो ये पूरे प्रोसेस को रेगुलेट करती है। वो क्लिनिकल ट्रायल कंडक्ट करने की परमिशन भी देती है, ड्रग इंपोर्ट करने के लाइसेंस भी देती है, ड्रग मैन्युफैक्चर करने का भी लाइसेंस देती है, ड्रग मार्केट करने का भी लाइसेंस देती है। तो सारी चीजों को रेगुलेट करने का काम है रेगुलेटरी एजेंसी का। इन्वेस्टिगेटर, ये बहुत इंपॉर्टेंट है। जो डॉक्टर्स क्लिनिकल ट्रायल करने में इन्वॉल्व है उन्हें हम इन्वेस्टिगेटर कहते हैं, और जो मेन डॉक्टर है उसे हम कहते हैं प्रिंसिपल इन्वेस्टिगेटर। अब ये आदमी पे पूरी जिम्मेदारी रहती है, पेशेंट को अगर कुछ भी हो जाए ये उसकी रिस्पॉन्सिबिलिटी है कि पेशेंट को पूरी तरीके से ठीक करे, कैसे भी करके। तो इन्वेस्टिगेटर का रोल बहुत ही ज्यादा इंपॉर्टेंट होता है। इन्वेस्टिगेटर ही डिसाइड करता है कि पेशेंट को ये जो साइड इफेक्ट हुआ है, वेदर इट इज रिलेटेड विद दिस न्यू ड्रग और समथिंग एल्स, तो इसके जो भी कमेंट्स है, जो भी डिसीजंस है, वो बहुत ज्यादा इंपॉर्टेंट है। और इन्वेस्टिगेटर जो बोलेगा, जो एविडेंस शो करेगा, यही एविडेंस के बेसिस पे क्लिनिक उस मॉलिक्यूल को अप्रूवल मिलता है या अप्रूवल नहीं मिलता है। तो ये सबसे इंपॉर्टेंट आदमी ये है इन्वेस्टिगेटर। लेकिन आपको भी पता है हमारे यहाँ पे डॉक्टर जो है उनके पास समय है क्या? आप कुछ बीमारी लेके जाओगे, 1 मिनट 2 मिनट ज्यादा से ज्यादा, और अगर आप अच्छे लग रहे हो तो वो 5 मिनट आपसे बात करेगा, उससे ज्यादा बात नहीं करेगा, निकाल देगा, बराबर है। 5 मिनट में कभी भी क्लिनिकल ट्रायल नहीं हो सकता, बराबर है। तो इन्वेस्टिगेटर को हेल्प करने के लिए रहते हैं एसएमओ। तो जैसे स्पॉन्सर को हेल्प करने के लिए कौन था? सीआरओ, वैसे इन्वेस्टिगेटर को हेल्प करने के लिए रहता है एसएमओ। सो वी आर अ एसएमओ कंपनी। हमारे पास जो क्लिनिकल रिसर्च कोऑर्डिनेटर है वो प्रोफेशनल्स है। ट्रायल कैसे करना है, कौन सी गाइडलाइन को फॉलो करना है, क्या करना है, इस विजिट में पेशेंट के साथ क्या करना है, ये सारा हेल्प कराते हैं वो इन्वेस्टिगेटर को, सो दैट इन्वेस्टिगेटर ट्रायल को एज पर प्रोटोकॉल और गाइडलाइन के हिसाब से कर सके। इथिक्स कमेटी एक ऐसी बॉडी होती है टोटली इंडिपेंडेंट, जिसका किसी से कोई लेना देना नहीं। वो एथिकल पर्पस देखती है पेशेंट के साथ। मतलब इस ट्रायल में अगर आप पेशेंट लोगे तो उसका इतना ब्लड आप विड्रॉ करोगे, तो इतना ब्लड विड्रॉ करोगे तो ये एथिकल है क्या? क्या सच में आपको इतने ब्लड की जरूरत है क्या? और अगर इतना ब्लड निकाल रहे हो तो पेशेंट तो मर जाएगा। तो हम ये ट्रायल को एथिकल क्लियर नहीं कर सकते। ऐसे डिसीजन लेने की जिम्मेदारी होती है एथिक्स कमेटी की। इस एथिक्स कमेटी में डॉक्टर्स भी रहते हैं, साइंटिस्ट भी रहते हैं, लॉयर भी रहते हैं, वकील भी रहते हैं, जज भी रहते हैं, ले पर्सन भी रहता है, मतलब जिसको कुछ नहीं आता, कॉमर्स आर्ट्स स्टूडेंट वाले, और सोशल वर्कर भी रहते हैं। यानी इनके पास से अप्रूवल लाना बोले तो बहुत बड़ा टास्क है, सभी लोगों को सेटिस्फाई करना पड़ता है। यानी हमारे स्टडी में साइंटिफिक पॉजिटिविटी भी होनी चाहिए, मेडिकल अप्रोच भी होना चाहिए, वो लीगल भी होना चाहिए, वो सोशल वर्कर को भी सेटिस्फाई करना चाहिए, और ले पर्सन को भी सेटिस्फाई करना चाहिए। तो ये इंपॉर्टेंट लॉ है। जब आप क्लिनिकल रिसर्च में एंटर करोगे कोर्स में, तब इनके बारे में आपको डिटेल में पढ़ने को मिलेगा। ये कुछ मैंने जॉब्स आपके सामने रखे हैं कि जब आप क्लिनिकल रिसर्च में एंटर करते हो, जैसे ऑपरेशंस में, तो स्पॉन्सर्स जो रहते हैं फार्मास्यूटिकल कंपनियां, उनकी साइड से डिफरेंट पोस्ट अवेलेबल रहती है। नीचे से ऊपर की तरफ जाएंगे, हमें ऊपर वाली पोस्ट पहले नहीं मिलने वाली, नीचे से ही जाना। जैसे सीढ़ी नीचे से चढ़ते ना, वैसे नीचे से ऊपर चढ़ना पड़ेगा। वैसे ही जो इन्वेस्टिगेटर साइड पे काम करते हैं तो नीचे से ऊपर चढ़ना पड़ेगा। डेटा मैनेजमेंट, फार्माकोविजिलेंस, रेगुलेटरी अफेयर्स में आप फार्मास्यूटिकल कंपनी जो स्पॉन्सर है उनके साइड से भी काम कर सकते हो, और डायरेक्टली गवर्नमेंट में भी काम कर सकते हैं, ठीक है। फिर मेडिकल राइटिंग, बायोस्टैटिस्टिक्स। ये सेशन काफी इंपॉर्टेंट है, लेकिन मुझे ऐसा लगता है कि मेरा टाइम यहाँ पे खत्म हो गया है। आपको ये सेशन के लिए आप मुझे नीचे मिल सकते हैं, हम नीचे में अवेलेबल है। हेल्प डेस्क के सामने हमारा स्टॉल लगाए। किसी को भी क्लिनिकल रिसर्च के बारे में और डिटेल में जानना है, इसमें क्या करियर ऑप्शंस है, क्या स्कोप है, हम आगे कैसे जा सकते हैं, तो मैं और मेरी कलीग श्रुति नीचे अवेलेबल है। आप हमें दिन भर में कभी भी आके मिल सकते हैं। अपार्ट फ्रॉम दैट हमारा, आप सबको शायद वो पॉम्पलेट मिले होंगे, छोटे छोटे लीफलेट, उसमें हमारा वेबसाइट भी और हमारा कॉन्टैक्ट नंबर भी है। आप कभी भी आके आपके जो भी डाउट्स है आप क्लियर कर सकते हैं। आपको आपके करियर के लिए ऑल द बेस्ट। आप लाइफ में सही और बेस्ट करियर चूज करें, इसके लिए मैं आपको क्या कहूं, मैं बेस्ट विशेष देता हूं। थैंक यू वेरी मच।
Thank you, Dayani sir, for gracing the audience and enlightening the knowledge over the drug delivery, discovery, development, clinical trials. Truly, your insight will be helpful to our audience. Thank you, sir. I request Ms. Pooja Dahikar ma'am to grace the stage and give small token of love and appreciation to Mr. Dayani sir for being a part of our conference. Thank you, sir. Thank you, ma'am. Here I declare the session is concluded. I give heartfelt thanks to Dr. Darshan Telange sir and Mr. Shubham Chinchulkar sir for gracing the conference with your presence. Now it's time to get recharged. We will take a quick break of 30 minutes and get resumed back by 2 p.m. There is a good opportunity to the pharma buddies to meet our sponsoring network at the ground floor. Stay tuned, stay connected. Thank you.